Welcome, dear online audience, to the data analysis for precision medicine meeting, day number 2. I'm very happy to have all of you around, as well as our live audience here in Paris, our institute. Today, this program is a little bit more computational than yesterday's. The session chair for this morning. We start right off with Kim-Anh Lê Cao. Anyway, I'm very pleased to have her here with us. Professor Lê Cao works in the area of computational statistics, in an area that has really been kind of growing on us a little bit. Biology has always been an important motivator for statistics. In fact, some of the first developments of those tools that everybody's using these days came from biology. Many of you know. We developed a statistical toolkit called Mixomics for, let's say, modern challenges like the computational facets of data analysis, when you have few observations and high-dimensional observations, and mixed data, mixed data from different sources, and we very much enjoy having her with us. Kim-Anh, the floor to you.
Thank you, Benno. So I'm going to share my slides. I'm going to give you a brief overview of the methods we have been developing in my lab for multiomics data integration. And to start with, I think this field, as Benno was saying, is quite cutting edge, but it also requires us to think in a very different way about data. And that has created the field of computational biology. We are basically, as a computational biologist, or I would say I'm a computational statistician, I work at the interface between biology and statistics. So biology, historically, is very qualitative, whereas mathematics, as you know, is very quantitative. And so bridging the gap between those two disciplines requires quite a lot of skills in terms of data analytics, but also, communication. And when I started that field, when I was doing my PhD, it was very clear that I needed to work really closely with both molecular biologists who generate the data, but also bioinformaticians who process the data in order to analyze some big biological data. And there were already a few challenges, and we're talking about, 12 years ago, 15 years ago, we already started to see that data would come from different types, and they were likely to be very big. We could already see that, our laptops wouldn't cope. And most importantly, I think I really started to realize that if I were only to rely on statistical methods, I would be unable to validate the results. And the reason being that the sample size were very, very small compared to the parameters, so the genes or the proteins that we were measuring. And so one way to validate those results was to actually rely on the biological interpretation. And then as the years came by, I also realized that it was a bit of a race all the time because new technologies were arising, and they're very disruptive in a way for us as computational people. I'm talking about, the advent of microbiome, so the study of all bacteria in a given environment, and then all the next-gen sequencing technologies, and then just recently, all the single-cell assays. So it's really important when you work in that field to always be a little bit at the cutting edge and work with the people who have access to those technologies. So I said that you need to shift the paradigm because, 20 years ago or even more, biologists were having a reductionist type of approach, where basically they would have a very strong hypothesis about one gene. They may knock out that gene, and then based on their hypothesis, and then you can do a statistical test on it. You can, on the data you have, so you can do a t-test or an ANOVA or whatever you want. But when we started to be able to measure thousands of molecules at once, so for example, 20,000 genes for a single sample, this is where the hypotheses are very unclear. And so it's really hard then to work out what is the test, what are we testing here, and can we actually answer the biological question, because actually there's no very accurate biological question that is asked to those data. So this is a holistic approach. I work with omics data, and most of the well-known omics data are transcriptomics, the study of transcripts. You have proteomics, metabolomics, but in there I also, include microbiome. I include every type of data actually that you can measure in a sample. And when omics data or omics experiments started to arise, we really thought that those different types of data should follow the biological dogma. And the biological dogma says that everything starts at the DNA, and then it's coded into RNA, and then it's translated into proteins and metabolites. So it seemed to be quite a straightforward relationship between those different types of data. But in fact, it's not. And so you can see that here within each omics or OM, you have different types of data you're measuring. So for example, if you're looking at the epigenome, you may look at DNA methylation, histone modification, chromatin accessibility, and you name it. So first, you have a lot of different assays that can measure each omics, and each of them will measure a specific type of molecule. And then you have a lot of crosstalk between those different omics. So it's not a linear relationship. You can see, for example, the transcription factors will have an effect maybe on the DNA. And so what we're trying to understand with those data is how do we explain the phenotype? How do we explain, why a person is sick or not sick for a specific disease, for example? That's really what we're trying to unravel here. And so historically, as I said, we started from a reductionist type of approach. We would look at, one gene or one protein across those individuals. And then about 15 or 18 years ago, we had the first omics data. So mostly microarrays and RNA sequencing data sets where you measure, hundreds, tens of thousands of genes on the samples. But you can do the same then for proteins or metabolites or whatever you have. And recently, in the past, 8-10 years, we started to look at multiomics. So this is where those experiments are performed on the same samples or the same individuals. And so here you're really trying to understand not only the relationship within each omic, so relationship between genes, but also the relationship across the different layers of omics. So is my gene related to those proteins, and are those proteins related to those metabolites, for example? And in fact, it's a bit of an analysis loop. So what we're trying to do in multiomics with this holistic approach is really we're trying to narrow it down to only a few different molecules that we think explain the phenotype. So that we can go back to a more reductionist type of approach. And this is where you then get the opportunity to validate your results in a wet lab, for example. So hypothesis generating in those experiments is key here. And there are different types of data integration methods out there. I'm going to focus my talk mostly on matrix factorization, and I will tell you what it is about. But just be aware also that you have different types of other methods. You have Bayesian methods, you have network-based methods, you have more simplistic approach where you look at or you analyze each omic layer independently, and then somehow you look at the overlap of the genes and between the data sets, for example. That also counts as data integration. So our hypothesis in multiomics is that that the cell response is triggered by a set of different molecules. And so you cannot adopt the one gene hypothesis paradigm, the reductionist approach, if you really want to understand a biological system in a holistic way. And so multivariate analysis is quite good to shift that paradigm, because instead of looking at a single gene or a single protein, we're going to look at a combination of different biomarkers. We are going to reduce the dimension of the data, because you can imagine now it's getting pretty big when you have different omics to deal with. And also this type of method is also very suitable for statistical data integration. So I'm going to explain a little bit those terms. But if some of you are interested in that field, just be aware that there's different types of expectations that you may have from that task. One is you may want to understand what is the role of each omics in a biological system. So for example, are genes able to explain my phenotype, or are proteins better? You might be interested in understanding the relationship between the different omics types, so between, say, genes and proteins and whatever you're measuring. You might be interested in what we call variable selection, which is identifying a very small set of markers that will give you more insight into the biology. Or you may want to also be interested in prediction. So you want to, based on the data you have, all these different types of information, you may want to say, well, I want to train a statistical model. And then if I have a new cohort, or if I have a new patient that give me their sample, I want to be able to predict whether that patient is likely to be sick, for example. And often I think a lot of people think that they're interested in everything. And in fact, the data may not answer all of this. And so sometimes all our approaches are data-driven. So they're driven by the data, and sometimes they may just answer a few of those points here. So just give you, I'm going to talk a bit more about the methods and what they're doing. So they call matrix factorization methods. And basically, the aim is that you start with a big data matrix. So imagine that you have, or it's actually rectangular, because you don't have a lot of samples or individuals. You may have, if you're lucky, you may have 50 or 100, but usually you don't have very big cohorts. But then you have a very large number of features. And so here, for example, my features are metabolites or genes. And so that's why I'm talking about a rectangular matrix. That's really hard to deal with computationally. So say if you want to fit a linear regression, it's actually quite hard to solve. And so instead of doing that, we're going to replace this big data matrix by new features or components that are going to aggregate the information from the data. So just imagine here that if you have one sample that is described by 20,000 genes, that individual lives in a 20,000 dimension space, okay, because it's described by 20 attributes. And so basically, by doing data reduction dimension, we are reducing this dimension to, say, here I have only 2 columns, so only 2 dimensions. And so those new dimensions are often called components. And basically, the way we build those components is we're going to aggregate all the information. So for example, for that individual here, I'm going to aggregate all the information of the 20,000 genes into something that explains the information in the data. What is great with those type of methods is that they are non-parametric, so they don't have any assumptions on the distribution of the data. They can handle missing values, they can handle zeros, which can also be a problem in a lot of statistical analysis. And they really aim, and the components really aim to explain what we call the variation or the information in the data. So I don't know if you've heard about principal component analysis. This is a very well-known multivariate methods that does dimension reduction. And we've been working on similar models called projection to latent structures because they're useful for data integration. So let's start first with PCA. It's exactly the same diagram as before, but I'm just going to give you a bit more details on what we do here. So we are going to extract from this data here 2 components. And each component is a combination of the P genes that I have here for each individual. But in order to combine these P genes, I need to be able to know how I can weight those genes. And those weights are actually indicated in what we call a loading vector. So that loading vector is going to say, oh, that gene is not important to explain the variance in the data, so the weight's going to be small. That gene is actually quite important to explain the variance in the data, so the weight is going to be large, and so on. And then you weight each of the values of the genes for that given individual, and you obtain one score. And so that's the score you would have for the first individual here. And you apply the same formula for the other individual. But that might not be enough. Maybe one component is not enough to explain the information from the data. So what you do is you look for a second component that is going to explain the remaining information that we haven't picked up from the first component. And so again, you have another loading vector, and you're going to recalculate those combinations. A variant that was proposed a while ago now is called sparse PCA. And basically what it does is that it shrinks, or it sets to zero some of the coefficients in the loading vector. And that means that when you calculate your component score, there's only a subset of those genes that are going to be taken into account in the calculation of the component. And so this is how you do variable selection, because basically you're saying, well, my first component is actually only based on 20 genes, and those 20 genes are important to explain what is going on in my data set. So yeah, there's three types of concepts here: components for the dimension reduction, loading vectors to build the components, and then feature selection. And so the key really is to define those loading weights or those coefficients. And so you do that with in an optimization procedure, where basically you ask to maximize the variance in the data. So this is, statistically speaking, this is how we then define the coefficients. And once we have the coefficients, we can calculate the components, and everything kind of flows from there. So that's version 1. So it's PCA, and it's really to explore the data. I don't know if you used it before, but it's a very useful data mining tool. The next version is projection to lesson structure discriminant analysis. And the difference with PCA, if you can see here, is that I have another type of information that is included. And basically, it's the information about the phenotype or the disease status of the individuals. And so here the aim is different, because you want to discriminate your sample groups. You want to know if there's any difference between my disease, my cases, and my controls, for example. And so, but the dimension reduction or the matrix factorization process is the same. So you will have components as well and loading vectors. It's just that they're going to differ, because they aim to discriminate sample groups this time, whereas before with PCA, we aim to maximize the variance in the data. And you also have feature selection, the same way I explained before, so you can actually identify now a set of markers that explain your phenotype. So one of the useful outputs, but there's many useful outputs from those methods, is to then plot the components. Just remember, each one component summarizes all the genes in the data set. And so I can plot component 1 versus component 2, and I'm going to see where my individuals fit in that space. So this is an example where it's a small round blue cell tumor data set. We have 63 samples and about 3,000 genes. And this is component 1 on the x-axis and component 2 on the y-axis. And so you have the scores of those individuals. And that's going to determine their coordinate in that plot. And it's a 2D plot. And so each dot here is one sample. And so you can start to identify similarities between a group of samples, for example, or dissimilarities between groups of samples, depending on where they are located on the plot. So that type of plot I show you, there's no color here because it's unsupervised. I never told the PCA that we wanted to, discriminate the sample groups or anything. I just put the data in there and see what happens. And on the right-hand side, you have the similar plot, but this time from discriminant analysis. So the components are different. And now you can also color your samples according to the tumor subtypes, for example. And so here you're doing a supervised analysis because you specifically asking to discriminate those specific groups. So that's really the difference between unsupervised and supervised. Unsupervised is more, let me have a look at the data and see what it tells me. And supervised is more, okay, well, actually I have additional information that I want to include in this analysis. So that's for one data set, okay? We looked at genes. I'm going to skip a few methods to go to the major one, which is integration of multiple data sets. And that method we call Diablo. And basically, it's also based on matrix factorization, but now you can see that I have several data sets to deal with. So for example, I have my genes, I have proteins, I have metabolites. They're measured on the same individuals here, but on different assays. And so the number of genes is different from the number of proteins that you have, and it's also different from the number of metabolites. And you also have a phenotype, an outcome or a response of interest. So here I can say, well, it's disease status, for example. So what we do is, again, we reduce the dimension of each of those data sets. So you can see here now we have the components. In those methods, the PLS methods, they're called latent variables. So just be careful the name change, but they are components, the same as what I showed you before. And then you have a loading vector for each of those components to calculate those components. And so the way you do the data integration is actually you ask those components. So here you have a set of components for the transcriptomics data set. And then you have a set of components associated to the proteomics data sets and so on. You constrain the mathematical model so that each component, the first component of the first data set, is highly correlated or covariant with the first component of the second data set, and so on. So basically, what you're trying to do here is find the optimal weights in your genes that are strongly correlated with the optimal weight of your proteins, and so on. So this is where the statistical integration happens. It's actually trying to find common information between those data sets. And we also do feature selection. So in fact, instead of looking at all the genes and all the proteins and so on, the components are only based on a subset of those variables. So you may identify, say, 20 genes that are highly correlated with 5 proteins that are highly correlated with 10 metabolites. And that's your signature, because we're also trying to explain the phenotype here. In terms of visualization, so if I show you a sample plot, as I showed you before, but for this kind of model, this is how it would look like. So you can plot each component associated to each data set. So here we have 4 data sets. It's a breast cancer data set. So you have mRNA, microRNA, methylation, and proteins. The colors here indicate the breast cancer subtype. And so from those plots, you can see that roughly, like if you look a bit far, we can extract similar information from those data sets. You can see that the basal samples are always on the right-hand side, and they seem to be separated from the other samples. If you look closely, you can see that some data sets are better than others to discriminate the sample groups. So for example, mRNA is very good at isolating or at identifying the basal samples. And then you see some discrimination here with the HER2 subtype and so on. Whereas if you look at, say, the microRNA, the difference between the HER2 in orange and the luminal B in green is not that cut off. And so this is what I was telling you before, that the data drive those models, and they already tell us what is the type of omic data that is important to discriminate the sample groups, if discriminating sample groups is your primary question for those data. So I talked a lot about samples and sample visualization, but I also told you that we do a lot of feature selection, and so you can actually extract those features and see how they correlate with each other. So this is a circus plot that we created, where under each quadrant here, you have the name of the mRNA that have been selected, the name of the proteins that have been selected, and the name of the microRNA that have been selected. And each line here indicates whether there is a positive correlation in brown and a negative correlation in black. And it just gives you some insight about the correlation structure of your data set. So from here, I can tell you that, in fact, there's not a lot of proteins that are correlated with the other types of omics, and that maybe between microRNA and mRNA, there's mostly a positive correlation. You can also look at that through heat maps, for example. So we have different types of visualization there to try to understand also the correlation between those omics at the feature, at the molecule level. And just one note on p-value, because Beno told me that you know what a p-value is now. It's actually not that relevant in multivariate data analysis. And primarily because when you've seen a p-value, you often have used univariate methods. So you test one gene, you get a p-value. That p-value tells you if there is a significant difference in the mean of the expression of that genes between different sample groups. But in multivariate analysis, it's very different because you're looking at a combination of genes and a combination of proteins. And so you may get a p-value for your whole model, but it doesn't tell you much about each of those molecules independently, because that's not what those methods are doing. They're trying to model all those features together. So yeah, you can have a p-value and it will tell you, oh yeah, your result is different from random. But it won't give you any insight into the role of each of those genes or those proteins, because we're trying to combine them in an optimal way to explain the phenotype and also to understand the correlation. Okay, so in the next 15 minutes or so, I'm going to give you 3 examples of application. The first one is what we call a bulk multiomic study. So just looking at samples from individuals. So here they are infants during the first week of human life. So we sampled those infants at day 0, day 1, day 3, and day 7. And then we ran different types of assays. This protocol is actually quite interesting because we could start from a very, very small amount of material. And then we generated transcriptomics, proteomics, metabolomics, and some other types of data. In the end, our data set was 30 newborns, and then we have samples collected at different times for those newborns. And we had 5 different types of data sets to integrate. And what's been also great with that study is we actually had a validation cohort from Papua New Guinea. So we could actually test our results in another data set or another study. And so what people do usually is they first look at each omic individually. And so those are the results of what happens in the transcriptomics data. So what we did is we just ran univariate analysis, and we counted how many transcripts were differentially expressed compared to day 0. So this is the number of transcripts differentially expressed between day 1 and day 0, and then between day 3 and day 0, and so on. And then you have a red line to tell you the genes that are upregulated or the genes that are downregulated. And what you can see is that as time evolves during that first week of life, the number of genes that are different from day 0 from birth increases. So you have really strong developmental changes. And we see the same with proteomics and metabolomics. So that's already one insight about what's happening. But the researchers were then interested in understanding what were the correlation between those transcripts and those proteins and those metabolites that were differentially expressed. And so they started to fit those networks where basically you calculate the correlation between all these different molecules, and then you visualize them into that network. And basically you can't see much because we call them the hairballs. There's so many correlations that are inflated. And the results are most of the time spurious because you estimating so many cross-correlation between so many variables. So this is the first part that we did, the Singleomics. Basically, we looked at every layer of information here, and then we calculated the Pearson correlation between the different differentially expressed molecules, and this is just a heat map of those correlation, and this is the network visualization. When we did something like using Diablo, for example, where we integrate all this information, you can see that there's a strong correlation structure that appears because those methods are designed to detect those correlations. And so then you can see different networks of different molecules that appear. And each network is related to pathways that are very specific or that seem to explain pretty well the first week of life in humans. And so what we found is we found some overlap of biological information between the two analyses, but also new insights. So, for example, we identified a gene called prostaglandin enteroprexide synthase 2 that we think is involved in differentiation. And so you can gain that way also additional insights with those analyses. So I forgot to say, I think, oh no, it's here. Yeah, so this paper has been published, and then we validated those results from the PNG cohort based on the Diablo model that we had trained. And so we asked, based on this molecular signature of those genes and cells and metabolites and proteins and so on, can we actually predict the day of life of that new cohort? So that's one way to validate the results when you don't necessarily, or you can't necessarily go back to the lab, is to have another validation cohort on the side. Another example is integrating microbiome and the omics from the host. So here, these are with researchers from University of Melbourne. They're looking at Huntington disease. And we've been already seeing that there was some dysbiosis in the gut microbiome in Huntington diseased mice. And those researchers are also interested to see whether we can understand or explain what we call the gut-brain axis. And one way to do that is to actually measure the plasma metabolites in the brain and see what is the effect in these mice. So we use microbiome data, so metagenomics of those mice and metabolomics of their brain when they are 12 weeks of age, and we're looking at wild type and Huntington disease group. So it's a very small data set. We only have about 18 mice. And yeah, based on, this is just the circuit that I showed you before, but this time for this study, so we identified bacterial species, we identified some genes from the metagenomics data, and these are the metabolites. And so mostly what we could see is there's a strong negative correlation between the metabolites and some of the genes and some of the bacterial species. And this is more like a cytoscape visualization of the results, just to look at this metabolite butyrate. We found that this metabolite was highly correlated with some specific species. We found that butyrate was decreased in Huntington disease compared to wild type. And that really, those kind of results really propose new hypotheses such as, is butyrate absorption impaired in Huntington disease mice. And so this is just an example to show you how, you can integrate different types of data with those models, but the analysis are mainly for hypothesis generation. And then from there, you need to kind of dig further to validate those hypotheses. The last example I want to show you is single cell, in case you're interested. This is an experiment where we're looking at mouse gastrulation. So we have gastrulation, we have different stages of gastrulation in mice and different lineage. And we use this fairly new-ish multiomic single-cell assay called SCNM-TSIC, where basically you can measure the transcriptome, but you can also measure DNA methylation and chromatin accessibility. And in fact, so here you can see we have about 800 cells, so not many, but it's still a single-cell assay. And you can see how those cells are divided into different stages of gastrulation as well as different lineage here. And those are all the data sets that we looked at. So as I said, you can look at the transcriptome. We have about 14,000 genes for these 800 cells. But then if you look at the methylation data, they can be broken down into different genomic contexts. So you can have the gene bodies, the promoters, and so on. So even though you think you have only one data set, you actually have many. And similarly for chromatin accessibility. So that leaves you with many, many data sets to integrate. This is some work that my postdoc did last year. Basically, we use a variant of Diablo, and those are the components that are shown for each of those data sets. So we have 13 data sets that we integrated, and each of those dots here now are cells. And they're colored according to their stage and lineage. So you can see, for example, that RNA defines stage and lineage pretty well, and that makes sense because we use RNA to define stage and lineage first. But then you can see how some methylation data are better than others to explain those phenotypic differences, and some of them are really not good, like this, for example. So that's on the cell dimension, and then if you look at the genes, if you look at the features that were selected, this is a Circos plot of the genes and the accessibility features that were selected and the methylation features that were selected. So you can start to see that there's a lot of negative correlation happening, not so much positive correlation and so on. So you can start to understand, if we have a valid hypothesis there based on the data that we generated. And finally, we then look more into, for example, the genes that were selected. So this is just violin plot of the expression levels of the genes that we selected across the different stage and lineage. And the gray box plot here represent the background noise. So it represents the expression levels of all the genes compared to the expression levels of the genes that we selected. And so what we found is that those genes were either switched on at the later stage, so that's this case here, you can see the expression starts to increase, or switch off, so they start, the other genes that were selected by the method were overexpressed, and then they go underexpressed. And then you can do some gene ontology pathway analysis on those genes, and to see whether the pathway that we identify makes sense or not. So in that case, they did make sense. But you have to imagine that you need to do all this type of analysis also on the other variables that you selected, so the chromatin accessibility, the methylation, and so on. So here I'm only showing you for the genes because it's easy to show, but it can become quite complicated. So just to show you that those methods are data exploratory methods. They're here to help you to narrow down your hypothesis and kind of direct you to the next steps of the analysis or the next steps in the validation. And I'll show you only a few methods here. I'll show you PCA, a bit of discriminant analysis, and Diablo. But in fact, we have developed quite a lot of those methods, and each of them are really designed to answer a specific biological question. And I think this is really important, is you can't just jump in and say, oh, let me just try a data integration method. You actually have to ask yourself, what do you want to answer from those data? Do you want to discriminate your sample groups? Yes, in that case, you need to choose a supervised method, or do you want to only look at one data set, or do you want to integrate multiple omics data sets, or do you want to integrate studies across different cohorts? So those are all fit into data integration. And yeah, so because all our methods are based on components, the outputs are the same. So I'll show you quite a few sample outputs, or the circus plots. We also have other types of outputs there to understand the samples and to understand the variables. So if you want to know more, you can have a look at our website. And we just published a book like this month that explain a lot about those methods and give you a lot of tutorials about how to use those methods in different case studies. And yeah, the take-home message really here is that when you enter that field, it's very data-driven, because we're trying to make sense of those data. I showed you that you kind of need to shift your analysis paradigm, because you're not in a reductionist type of approach now. You're more holistic. You want to generate new hypotheses. It doesn't mean that you can't apply biological or include biological knowledge here. You can, whether you do it at the end of your analysis. Sometimes you can do it at the start as well in those methods. And if you're interested in computational biology, basically, you may deal with two types of people: some who are more computational, some who are more biological. And I think it's important now in the newer generation that we have a blend of those disciplines and expertise. And I'll show you the example with Mixomics. It's one of the computational tools that is available to the community. And it's really important to also make those contributions to advance knowledge, but also sharing data is really important for that field, so that we can test our methods on different data sets. And so that's it. Those are all the people I would like to thank. I've been working with a lot of collaborators along the years. And also we have a lot of feedback from our users, from our Mixomics users. Thank you. I can't hear you. No.
The audience, can you hear me?
Yes, I can.
Okay.
I can hear one person.
Maybe there is a technical problem locally. In the meantime, if there are questions from the audience, can you put it on the chat, please? So there was one question: matrix factorization-based methods may not be able to perform inductive inference. I wonder how we can find a solution to that.
There might be, it depends on the type of methods you're looking at. I think some of them can do inference. So you can look, I think, at, well, I don't have specific examples, but you should, maybe you can look at latent factor models, because those models are more, well, they're modeling methods, but of course and you can use the latent factors for visualization as well, but they're a bit more complicated to run. And of course, as soon as you start to do inference, you start to make a lot of assumption about the distribution of your data as well.
Yeah. So there is another. So Vanessa Vermeersen, she wonders for compositional data like microbiome data, are there extra precautions that need to be taken into account? For instance, the application of Diablo that you were showing?
Yeah, so it's a very controversial topic,
Yeah.
because basically every time you look at the abundance of one bacteria, it depends on the abundance of the other bacteria that you've seen in that sample. And so there are quite a few tools that try to, statistical methods that try to deal with that from the, attack the jugular with that. But in our case, we use centered log ratio transformation. So it's a more pragmatic approach where basically your data are constrained because of that compositionality, and using that transformation actually breaks the compositionality structure. It's not 100% perfect, but it's working well enough. And so what you should do is transform your data beforehand before you then include them into Diablo or any other methods you want, basically, that doesn't handle compositionality internally.
Maybe along the same lines, also when you're doing a dynamic analysis with microbiome data, because I mean you're interested in correlations again, also from the time perspective. So is
it's a very good question, and we're trying to understand what is the effect of compositionality in longitudinal microbiome studies. Because can you actually really compare what happens between day 1 and day 2 and day 5 when you have different compositionalities? And if you use this transformation, is that enough to actually link those time points together? And we don't really have an answer yet. So at the moment we do CLR transformation as well, but we do realize that there are limitations in the interpretation we have. We may have now more PCR data that gives us, the absolute counts, and then maybe we can check then if the method is doing well or not.
Yeah, we're also working a little bit in that area. I'm, by the way, the coordinator of TranSYS.
So if people are interested in longitudinal microbiome, longitudinal omics integration, we have a few tools, and it's implemented in Thymomics. So it's for Thymomics. Yeah, you can have a look there.
I see one more question. Oh, Benno, can you take over?
Oh, yeah, cool.
Okay, there are two more questions
in the room here with the ESR. Thanks, Christelle, for filling in. Sorry about the hiccup. Do we have questions here in the room? Bechnam, did you type a question? Yeah, you can come here and ask it. Look in the camera.
Hello, thank you so much for the nice presentation. I'm Behnam, ESR 8. My question would be, I was considering that you are combining different data types together, and do you consider weights for each of them to combine to give weight to one specific data type more than the other?
Yes, yeah. So I haven't talked about the parameters in Diablo, but there's one where you define the amount of correlation you want to maximize between data sets. And so you can just say, oh, I don't really care, I want full maximization, and in that case you put a weight of 1 between pairs of data sets. But sometimes you can say, well, actually, I think my plasma metabolites are really not correlated with my whatever I'm looking at. And in that case, you can change that weight as well. So again, it's a data-driven process. So you kind of have to try, and you kind of have to explore the data beforehand to try to find or set those weights.
So it's more intuitively rather than just use optimization methods to reduce a specific cost function to make sure that the weights are more.
What we do is we evaluate the performance, so we can evaluate the classification performance of the model, and that may already give you an indication of whether those data sets are the correlation is good or not. So, but often you have to kind of run the whole model and try to work out what went wrong or didn't go wrong, and then kind of go back and start again.
Okay. Thank you so much.
Are there other questions in the room? I would have one maybe about you know, your goal to get closer to the biology, etc. There, it does occur to me that when I look at a lot of biology, people have things like feedback loops. You've had them in your slide with proteins act, transcription factors act back, and there are so many classical feedback mechanisms known and non-linearity known. So I'm wondering how you think about the limitations that you reach when you use these methods that make these linearity assumptions. Are you at all worried about that, or what is your thinking about that when you go to biologists and apply a linear model to a problem where maybe you don't know ahead of time whether?
Yeah, so I think that rejoined the question from Emil, who asked what is the limits of Mixomics. And yeah, so our methods are linear. And linearity is the easiest thing to explain to a biologist, because as soon as you start to be non-linear, it's like you have to say, well, it's actually the expression of your gene to your gene, you know, to cubic, you know, square expression values of this, and they start to really struggle because they can't really work out what is important or not. But it's like, and so what we do with those exploratory tools is we're really trying to explain what is the most obvious, and the most obvious would be linear. But I do believe that biology is not linear. But I think you first need to use those tools as a filtering process, where you say, tell us what are the few genes and bacteria that I should focus on before I can move on to more advanced, sophisticated mathematical models. So for example, in microbial communities, we're starting to look at Lofka-Volterra models, where you can start to infer causal relationships between, you know, that bacteria and a set of bacteria. And so that's, you can only do that when you have already a small pool of candidates. So there are nonlinear methods in that field, like you can do kernel methods, for example. But just interpretation is really hard. And I think if our audience is mostly biologists or computational biologists, I think it's more strategic to give them a linear solution first.
Thanks. Follow-up question, because you mentioned the word causal relationships. That's another thing. These models that you're using, they seem to tend to be based on the idea of correlation. Are there tricks? I mean, how do you go? And the biologists make their qualitative diagrams with a lot of directed arrows, not just clusters that move together, etc. What are your thoughts on that? Maybe also from a method side, is there need for methods development?
Yeah, but it's hard. So I don't know if anyone has solved it yet, because what we find is that the assumptions we have in biology don't match the data that we generate for some reason a lot of the time. So, for example, in methylation, we expect to see positive relationship between gene expression and some specific methylation regions, and we don't find it. And we are unable to say if it's because of our methods or if it's because of the technology that currently is still not accurate enough, or is it because our assumption is completely wrong? So I think, yes, there is space in that area, but I think we're very far away. And you can see in single cell how people are all about hypothesis generating now, because they realize that there's no way they can actually, infer anything really strong from the data. We're still in the stage where we don't know what the cell types of the cells are. There's a question from Leon as well, who asked if the data need to be normalized before you put them into the methods, and the answer is yes. So you need to process them and normalize them according to what makes sense for the specific omic data type you're looking at. So we haven't really included that in the package because, we're looking at so many different omics. There's no way we can propose a normalization method for each of them. Also, one mistake that users do is they try to fit all their, say, 500,000 accessibility regions in their model, and that's going to take a while to run. So it's actually quite important to filter the data as well, because we know that a lot of those features actually don't change whether you're sick or not sick. And so you can just calculate the variance, for example, and just keep the features that have the highest variance across your samples, and not be afraid basically of getting rid of quite a lot of data, because we know that it's extremely noisy.
Thanks a lot, Kim-Anh, for joining us today. We've learned a lot about the space that you've opened up for us, and have a nice evening there in Melbourne. And Elva-Maria Novoa-Del-Toro is the full name. Is working on other ways of integrating data that Kim-Anh actually mentioned. If you remember the one slide with different approaches that include matrix factorization, another of these approaches was networks. Networks typically refer to connections that you get when you look at which molecules, which components of the biology interact. And it's another way of bringing in multiple informations about biological system, and you have the observation that Kim-Anh was talking about on the transcriptomic, proteomic, metabolomic level, etc. But then explicitly including maybe knowledge about the molecules that interact. And Elva is traveling today. She won't be able to give us a live talk, but she sent us a recorded talk and will be around for questions after this.
So I would like to thank the organizers of the TranSYS Winter School for inviting me. And thank you for attending to my talk. I hope you enjoy it. Today, I will present the project that I developed during my PhD, which I did at the University of Aix-Marseille, and in particular at the Marseille Medical Genetics Lab in the team of Dr. Anaïs Boudou. And this project is about detecting active modules in multiplex biological networks. I will start with a very brief introduction of the biology that is the basis of this project, and then I will explain you in detail what the method that I developed does and how it works. So in general, we can say that all the cells that we have in our body have the same DNA, but however, we have very different cell types, which have very different structure and very different function. So for instance, we have brain cells, heart cells, blood cells, etc. And what it is responsible for such variety of cell types is gene expression. It means that in every cell type, there will be a combination, a particular combination of genes that are active or being expressed as compared to a different type of cell. And we also know that the gene expression is affected in disease states. This means that the level, it's not that it can happen that cells from a patient have a different combination of genes that are being expressed, but also the level of expression of those genes can vary in patients as compared to controls. And given that when a gene expresses, it generates RNA, we can estimate the gene expression by measuring the amount of RNA present in a sample. There are different techniques to do so, but two of the main ones are RT-qPCR and RNA-seq. Inside the cell, there are different molecules that are generated, as I said, when the genes express. So we have, for instance, proteins and different types of RNA, and all these molecules do not act isolated to accomplish the cellular functions, but they rather interact, forming complex networks of signaling, collaboration, and communication. And we can represent all of these interactions with networks, where the nodes will represent the molecules, and then two nodes will be joined by an edge if the corresponding molecules share a relationship. So for instance, in a protein-protein interaction network, we will have that the nodes will be the proteins, and then two nodes will be linked if the corresponding proteins can interact physically. And thanks to the guilt by association principle, we know that all the molecules that participate in the same biological process tend to lie close to each other in this type of biological networks. We can therefore say that biological networks are the basis of biological processes. So I developed a genetic algorithm in order to analyze gene expression and biological networks to find the regulated cellular processes between two conditions. And in particular, I'm interested in studying the differences between patients suffering from a genetic disease as compared to healthy controls. So as I mentioned, I have two data sources. And the RNA-seq expression data can be used to find the set of the regulated genes. It means that we will use statistical methods to compare the expression levels of patients as compared to controls, and we will find those which are, for instance, upregulated in patients or downregulated. And then we can use this list to find the enrichment of annotated functions. And this implies that we will look for the annotated function of each of the genes that are significantly differentially expressed, and we will see if there are any functions that appear more frequently than what would be expected by chance. And this sounds very good in theory, but in reality, we do not have an annotation for all the genes. It means that we do not know which function is involved in every single gene. And also, this type of enrichment analysis is biased to well-studied functions. And the second data source that I used are biological networks, which, as I said, contain the information for cellular processes and also pathways and complexes, but biological networks are generic, which means that only by looking at the biological network per se, if it has no other context, we cannot know which cellular processes, pathways, and complexes are involved and or affected in the disease under study. But the good news is that we can integrate these two data sources in order to overcome their individual limitations and to have access to the active modules, which are highly connected subnetworks with an overall deregulation. And the active modules can help us go from the deregulation at the gene level to the deregulation at the level of biological processes, and this is thanks to the guilt by association principle that I already mentioned. One important characteristic of active modules is that not all the genes have to be significantly differentially expressed, and this relaxes the constraint of classical functional enrichment analysis. A real biological network looks more or less like this one. We can have thousands of nodes and up to hundreds of thousands of edges. And in this figure, every subnetwork highlighted with a different color could be an example of an active module. And the identification of active modules is an NP-hard problem, and therefore there have been different approaches and heuristics that have been proposed to solve it. The three main ones are based on greedy searches, simulated annealing, and genetic algorithms. Greedy searches are like hill climbing. It means that given a set of options, we will always choose the one that is the best, the one that maximizes the score. And Pinnacle set is such an algorithm. So let me explain you greedy searches with a toy example. Let's suppose that we have this little network and we want to select a connected subnetwork from it. So we see that inside every node we have a number, which is the weight of that node. And what we want to do here is to select a connected subnetwork. And we will start at a node that we will choose randomly, and then we will add one node at a time. But the restriction or the constraint is that in order to add a new node, its weight has to be higher than the previous node. So let's suppose that we start here, since we want to find a selected, a connected subnetwork, so these are the three options that we have. The one with the best weight is this one, and its weight is higher than the previous one. So we can add it. We continue the greedy search. Now these are our three options. This is the best one. Its weight is higher than the previous one, so we also add it. But then we reach to this point where none of the weights are higher than the previous one, so we cannot add any other node. Therefore, the greedy search stops. But this is what it's known as local optima, because the node that had the highest weight could not be reached. So we got trapped in, as I said, a local optima. Now, the second approach that I will explain you here is simulated annealing. And this is also like hill climbing, but with random choices. It means that if we have a favorable move, we will always keep it, but we can also accept unfavorable moves, and this will depend on the value of the temperature parameter, which is usually high at the beginning, which makes it more likely to keep unfavorable moves. And then the temperature parameter, the value for this parameter decreases as the search progresses. It means that at the end of the simulated annealing process, it would be very unlikely that we choose an unfavorable move. And J-active modules is such an algorithm. So with the same two examples that I showed you before, where with the greedy search we were already trapped and we could not add any further node, simulated annealing can help us escape this local optima by choosing an unfavorable move. In this case, it's this node, which will not maximize the score of the subnetwork, but that also has the potential to help us reach the node that had the maximum weight. So this would be the global maxima. The last approach that I will explain you here are genetic algorithms, and the method that I developed is also based on a genetic algorithm. So in this case, it is about emulating the natural evolution of species, and we will analyze a population of individuals simultaneously, and every individual is a potential solution to our problem. So in the case of active modules, every individual will be a subnetwork, which is potentially an active module. And the best individuals will be tuned, whereas the worst will be discarded. And the population will evolve until it converges. And cosine is an example of a genetic algorithm that looks for active modules. So I just mentioned three different approaches and three different algorithms that apply these approaches. But there are several out there. But in general, only very few of them consider the density of interactions. And this is important because for a method that only considers the weights of the nodes, these two subnetworks would be equivalent, whereas we see clearly that the second one is densely connected, which would imply that it is more likely that the corresponding molecules are involved in the same biological function. And also, in my opinion, the most important limitation that current methods have is that they can only analyze single networks. But we nowadays have access to a variety of biological networks, and it would be nice to have a method that analyzes them simultaneously. So having these limitations in mind, I developed MOGAMOON, which is a multi-objective genetic algorithm that has two objectives to maximize: the average node score and the density of interactions. So the average node score represents the overall deregulation of the subnetwork, and the density is the number of edges that there are inside the active module. So these are the objective functions, and I will now show you the flowchart of MogaMoon, which is composed of 10 main steps. And MogaMoon is based on NSGA2, which is also a multi-objective genetic algorithm. And this was proposed more than 20 years ago, and is an algorithm that has been widely used, and it has been shown that it works pretty well. So in this case, I adapted it in order to be able to process networks. I will explain you each step of this flowchart in detail, and then I will clearly state in which steps I adapted NSGA2. So MogaMoon starts by generating an initial population. This is a random process where 100 subnetworks will be generated, as I said, randomly. Each of these subnetworks is a potential solution, and in order to generate one subnetwork, I pick randomly one significantly differentially expressed gene, and I use it as a seed for a depth-first search. And this is one of the steps that I adapted from the classical NSGA2. Then in the second step, I evaluate the population. So using the objective functions that I showed you before, I will calculate for every subnetwork the average node score and the density of interactions. Once the population is evaluated, I will then rank it with respect to their values of the objective functions. And the technique that I use is called Pareto dominance. And in this case, if you have a look at this figure, every dot represents a subnetwork, so an active module in this case, and it is plotted in the coordinates that correspond to its values to the average node score and the density of interactions. And we say that a subnetwork dominates another one if its values for the average node score and the density of interactions are higher or equal than the second subnetwork, and at least for one of the two objectives, it has a better value. So that the value of either the average node score or the density of interactions is superior. And thanks to this Pareto dominance, we will separate the population in what is known as Pareto fronts, which are represented here with different colors. And the best individuals are in the first Pareto front, and they have a rank equal to 1. So now we have a population that we will use to generate a new one, but we need to select parents first. And MogaMoon does it by tournament of size 2. It means that we will select randomly from the full population 2 subnetworks. We will compare them, and the one that has better values, so that it's better ranked, it will be selected as a first parent, and we repeat the process to select a second parent. Once we have chosen 2 parents, we are ready to apply the genetic operators of crossover and mutation. So during crossover, 2 children are generated, as you can see here in the figure. Each child will take a part of each of the parents, and then during mutation, there will be nodes that are either added and or removed from the children. And this is another step that I adapted from NSGA2. And thanks to these operators, I make sure that all the subnetworks or all the individuals remain connected through all the evolution process. So now we have 2 children, but we need a full population of them. So we repeat the selection of parents and the crossover and mutation several times to generate a full population of children. So in this case, we will repeat the process 50 times. And now we evaluate the children that we just generated using the 2 objective functions, as I told you before. And then we combine the 2 populations, so the initial population and the children, so that we have a joint population of 200 subnetworks. And at this point, I added an extra step where I replace the duplicated individuals with random ones in order to avoid premature convergence and also to assure that the diversity will be maintained in the population. Then, so we have 200 subnetworks in the joint population, and we will rank it with Pareto dominance, as I explained before. And in the step number 9, we will select a new population because the size of the population has to be maintained during the evolution process. So we have 200 subnetworks at the moment. We need to choose 100 of them. And in this case, I do it with elitism. It means that I will take the top 100 subnetworks, so the best 100 subnetworks out of the 200, and I will replace the population with them. So now we completed one generation. It means that we started with one population and we used it to generate a new one, which in theory will be a little bit better than the previous one. So with each generation, the population will improve a little bit, and we will continue this evolution process until reaching the stopping criteria, which in my case is a fixed number of generations, which is equal to 500. Now I will show you how the performance of Mocamoon compares with state-of-the-art methods. So in this case, I will compare it against Binacle set, cosine, and J-active modules, which represent the three main approaches that I just explained you. And in this benchmark that we designed, that was actually proposed by Batra et al. In 2017, the idea is to generate one active module and to see how good the methods are to retrieve it. So the active module is represented here by this yellow subnetwork, and it is composed by 20 foreground genes which are connected. And we generate artificial expression data in order to highlight this active module from the rest of the nodes, which will be the background genes. Okay, so for this, I have two different protein-protein interaction networks which have different characteristics, as you can see here. And I also used two different data sets. So the first one, the distribution of the expression follows a normal distribution, but it's just that the mean of the distribution of the controls as compared to the patients is different. And you can see that we do not have only 20 significantly differentially expressed genes. We have almost 500. And this is because this is only considering the p-value and not the FDR. And the reason why I decided to do it like this is that I wanted to make it more challenging for the methods to find the active module. And in the second case, I sampled the expression data from TCGA breast cancer data. And in this case, I do have only 20 significantly differentially expressed genes because the expression data from the patients was sampled from the set of significantly differentially expressed genes in breast cancer and the controls from the non-significantly differentially expressed. Okay, so these are the results of 30 runs. In this case, every dot represents an active module that was retrieved by any of the methods, and the triangle represents the foreground genes, so the active module that we were looking for. At a first glance, you can say that Binacle set is the one that found the best results because the values for the subnetworks that it found are superior in both average node score and density of interactions. But when I had a deeper look at these subnetworks, I found that they were very, very small, starting at only 2 nodes. And if we are looking for the deregulated cellular functions, and we have a look at only 2 nodes, even if they are connected, it is very unlikely that we can really find out which biological function is behind that interaction. So discussing with our colleagues who are biologists, they considered, and also by looking at the literature, they considered that 15 was the minimum acceptable size of a subnetwork, where they can have enough information to see if the corresponding genes were actually involved in the same biological process. So what I did was to remove all the subnetworks that had less than 15 nodes, and at the end you can see that Mokamoon is the one that finds the best results. So in the case of Mokamoon, I only allow subnetworks of at least 15 nodes, and the other methods do not have this option. And the second experiment, using the second PPI and the expression data that was sampled from TCGA, you can see that we have more or less the same pattern as before. But again, when I remove the small subnetworks, you can see that Mokamoon is clearly the best. I also compared the methods in terms of their F1 score, which allow us to see not only how good the methods are to actually locate the foreground genes, which are the true positives, but also to avoid picking background genes. In this case, these are the false positives. So the reason why J-active modules cosine and Pinnacle Z have a lower F1 score as compared to Mokamoon is because even though all the four methods found all or most of the foreground genes, these three selected more background genes than Mokamoon, and this is why the F1 score is lower. So going back to the definition of the objective functions that I showed you before, this formula to calculate the density of interactions can only work when you have one layer or one network. But as I said, we want to analyze several networks at the same time. So the idea is to be able to analyze a multiplex network where we will have several layers, where every layer is a different network, and we have the same set of nodes in all the layers, and every node is connected to itself across the layers. So for the results that I will show you later, I used a multiplex network composed of 3 layers, and we have the same set of nodes, as I said, and the layers represent a protein-protein interaction network, pathways, and correlation of expression, and I will refer to this layer as co-expression. And you can see that they have a very different number of edges. So the topology is very different in each of these networks. So in order to be able to analyze a multiplex network, I adapted the density of interactions so that it is calculating the density of the subnetwork as compared to the density of the full layer. And this also allows to give more importance or more weight to the edges that come from the networks that are not that dense. And we decided it like this because the edges in which we can trust more are the ones that are experimentally obtained, that have been curated, and that we are sure that those interactions really exist. And those are the protein-protein interactions. And we also have a very, very dense layer, which is the co-expression, but this is also the noisiest one. So now I will show you how I applied Mokamoon to analyze real use cases. So I will show you the results for the analysis of a rare genetic disease, which is called FSHD, and also to breast cancer. So FSHD stands for facioscapulohumeral muscular dystrophy, and this is, as I said, a very rare genetic disease. And for this, I had 3 different data sets, which come from different cell types. And you can see that the number of significantly differentially expressed genes is different in each of these data sets. So I analyzed all of them using Mogamon. I ran the algorithm 30 times, and these are the summary of the results. So the first thing that I want to highlight is that even for those data sets that had very few significantly differentially expressed genes, Mogamon was able to find active modules. And in this case, the number of genes that are inside those active modules is superior than those in the set of significantly differentially expressed genes. And in the case where we have a couple of hundred of genes, of course, it also finds active modules, but in this case, it decreases the amount of, let's say, like important genes. These are examples of two of the active modules that Mogamon found, and you can see that they are different. So, okay, I will explain you what the colors mean. In the case of the nodes, the colors show either if the corresponding gene is upregulated, if it is red, or if it's downregulated in patients as compared to controls, if it is green. And then the significantly differentially expressed genes are marked with a black border, and the color of the edges changes with respect to the layer where that edge came from. So the active modules that Mogamon finds are very different. And here, like for instance, this active module caught my attention because the more the regulated genes, these 5 that are highly upregulated, are only connected by co-expression edges, which means that these genes do not interact physically, so they don't have protein-protein interaction edges, and they do not belong to the same pathways. So at the end, it is unlikely that they are involved in the same biological process. So there will be some cases where you will have a combination of edges, so coming from all the layers, and in this case, it is more likely that the genes are involved in the same biological process. And I also used Mogamon to analyze breast cancer data. So in this case, I obtained it from TCGA, which is a repository that is available on a web page from the USA. And they have available different gene expression, so RNA-seq expression data from different tumor stages. And in general, we have the gene expression data from more than 1,000 patients, and they are a little bit more than 100 controls. And here is the list, so the summary of the number of significantly differentially expressed genes if we take into account the log-fold change and the FDR. And here, for instance, if we compare all the patients disregarding the tumor stage as compared to controls, we have more than 7,000 significantly differentially expressed genes. And even if we consider only a single tumor stage, so if we take some of these 1,000 samples and compare it against the 100 controls, we will still have thousands of significantly differentially expressed genes. So I wanted to see what were the difference between patients and controls. So I started with all the patients disregarding the tumor stage, and the only active module that Mogamon found was this one. And you can see that it is highly connected. So there are a lot of edges coming from the 3 layers, and you can also see that it contains a lot of significantly differentially expressed genes. So in this case, there are 10. And this active module is associated with the DNA replication process. And of course, it is logical that in cancer, there is a deregulation in the DNA replication, because the cells are growing very fast. They are being reproduced very fast. but I also wanted to know a little bit more. So what I did was to take only a single tumor stage and compare it with the controls, but it seems like this active module is really the most deregulated one, so it was the only one that was being retrieved. And you can see here there are 3 different tumor stages and then all the patients together, and there are more than 1,000 common genes. So yes, this seems to be like the only active module, or the most active module in this case. But I still wanted to know more. So I decided to change the question, and instead of wondering what was different between patients as compared to controls, I wanted to see what was different between tumor stages, so between patients. And in this case, I analyzed, for instance, the tumor stages 2B as compared to 1A, which have more than 600 significantly differentially expressed genes. And in this case, I did found more active modules. And here you can see 2 examples. And of course, we still find the DNA replication, but we also find other active modules which are related to other biological processes. So in this case, in this example, we have this one that is related to signaling and cell communication, and it has a lot of pathway links and also some protein-protein interactions. And you can see that this one has no correlation of expression. So this module it's very interesting. So each of these active modules, it's a different path that we can then explore further. And here I analyzed 2 other different tumor stages, and in this case there were again more than 600 significantly differentially expressed genes, and Mokamun found 14 active modules, which included 21 of the significantly differentially expressed genes, but other 76 that were not significantly differentially expressed. And so you can see that again we have the DNA replication, but we have other functions that could be potentially interesting, such as tissue development, cell death, cell differentiation, etc. So as a conclusion, I would like to say that Mokamun finds different alternative paths that we can explore. So each, as I said, each active module is a different path that we can then analyze to see what was going on in patients, why this process was deregulated, to find out if it really belonged to a single biological process. And Mokamun, it's really like versatile in the way that if there is too little information, so if you have a data set where there are very few significantly differentially expressed genes, Mokamun can help you to complement this list in order to find some complementing genes that, even if they are not significantly differentially expressed, they are potentially interesting because they interact a lot with the genes that are differentially expressed. And the other way around, if you have too much information, like in the case of breast cancer, where we had thousands of significantly differentially expressed genes, Mokamun can help you to filter this list to keep only the most interesting ones. And another important characteristic of Mokamun is that it adds biological knowledge, which is the contribution of the biological networks. So it adds biological knowledge to the expression data. And importantly, Mokamun can be applied to any genetic disease. So this is why I showed you the results of not only a rare genetic disease, but also an unfortunately very common one, such as breast cancer. And to the best of my knowledge, Mokamun is the first method that is adapted to analyze multiplex networks, so several networks simultaneously, and it is also available as a bioconductor package. So before finishing the talk, I would like to tell you very briefly about the project that I am currently working on. So I am part of a consortium that gathers 4 teams, 2 from Germany and 2 from France. And in this case, we analyze the metabolism, so either of C. elegans, plants, or human. And we have metabolomics data that we can use to build several different experimental networks. So we can use these metabolomics to, for instance, calculate or obtain a network that represents the mass differences between the corresponding metabolites, or we can also find one or calculate one that represents the correlation between those metabolites. And we also have one that is knowledge-based, which is the genome scale metabolic network. And the idea is to build, as you can see here in the figure, to build a multi-layer network where we will have all the networks that we built from the experimental data, and we will also have the knowledge base, so the genome scale metabolic network. And the reason why we want to do this is because all these layers are complementary. So we can use, for instance, the genome scale metabolic network to filter spurious connections from the experimental network. And we can potentially also use the experimental layers, for instance, to solve the gap filling problem. And for this, with this, I mean that there are some metabolites that might be missing, so some reactions and metabolites that might be missing in the genome scale metabolic network that we actually observe when we do experiments. So the idea is to use all the information that we can gain from each of the layers and use it to complement the other layers and to filter the information. But here, one of the most challenging tasks is to find the correct interlayer connections, because to determine to which metabolite a node from an experimental network corresponds to, it's not that straightforward. So in the future, it would be interesting to show you the advances of this project, but for the moment, this is ongoing work. I would like to thank all of the people that were involved in the development and the writing of the paper of Mogamoon, and also all the people with who I am currently working with. And thank you for your attention. And now I will join you on live to answer all the questions that you may have. See you soon.
All right, thanks, Elva, for this nice talk. You have joined us online. Right. Yeah. I think you can unmute yourself.
Yeah.
Welcome. Can I ask for questions in the audience here or elsewhere? Please step up. Federico, question?
Yes, sorry.
Over there.
Over there, okay, perfect.
You see her behind you. Yes.
So my question is that in the objective function, there are two parts, and one of the parts is the density part, in which the more dense the submodule, the better. But,
sorry, can you please repeat? I'm not getting it. I will just turn my camera off.
Okay. Okay, what I said is that in the objective function for the Pareto function, there is a part which is density based, so the most dense, the most connection we have in the submodule, the best.
Yes.
But that is made with some sort of proportion with the maximum amount of edge that you can have in the module. So my question is, how can you do to make sure that this does not penalize too much bigger modulus? Because that way the denominator becomes bigger and bigger.
Yes. Well, actually, that is the work of the genetic algorithm itself. When it optimizes at the same time the average node score and also the density of interactions. Can you hear me well?
Yes.
Okay, So when it optimizes simultaneously these two objective functions, it also makes sure that the module does not grow too much. And actually, we had the opposite problem. Because it seemed like the smaller the module, the more, like the higher the values for the objective functions were. And this is why we had to, at the end, set a minimum size for the modules, because yes, we actually had the opposite problem. So this is why in some of the results that I showed you, for instance, Pinnacle set, since they don't limit the minimum size of the modules that they allow, they ended up with very, very small modules.
Thank you. There's more questions here. Please step up. You can step up, stand in line. Then we can switch more quickly. Thank you.
Hello, thank you for a nice presentation. I'm Behnam, ESR8. My question would be that some genes have the inhibitory effect. So if they are overexpressed, then it means that our module will decrease in terms of its activity. So so it seems that it would be vice versa. So how do you think about this issue, and is there a method is able to solve this issue as well?
Inhibitory, do you mean like post-transcriptional regulation?
I mean, okay, we are looking at the genes whose expressions are increased. So, and they say, okay, this module is activated if the, for example, if it is the gene expression, and if the if the expression of the gene is increased, and we have a module whose all the genes are increased in gene expression, then we would say, okay, this gene module is active. But some others among these genes, some of them have the inhibitory effect. It means that if they are overexpressed, they decrease the activity of the module. So so yeah.
Yes, I understand now what you mean. Thank you for the clarification. Well, the thing is that whatever that it's different as a biological process itself, and that would differ between patients and controls, would be interesting for us. And the term active module does not mean like all the genes that belong to this module are participating to like activate a function. It's just a name because the genes are overall deregulated, but we are actually not looking for only upregulated genes. It can be a mixture of upregulated and downregulated. We are just looking for the regulation like overall in the whole subnetwork. And then even if the genes that are involved have an inhibitory, sorry, I'm in the airport and they are giving an announcement. So even if the genes that are involved in the active module have an inhibitory function, I think that would be interesting for biologists to further analyze what is going on with patients. That why are these genes active if they were not supposed to? So yes, I think it's anyway interesting. But yeah, I think maybe the confusion was with the term active module.
Yeah, okay. Thank you so much.
Thank you.
Hello, thank you for the nice presentation. My question was that, how you involve the role of temperature and calculating the scores, and how does it change in different scales when the temperature changes?
The temperature is a parameter that I don't use because this is from simulated annealing, and this comes from J-active modules, which is another method, and it's a parameter that it's being its values update automatically. I mean, these are calculus that depend on the values of the active modules that are being retrieved, but this is not part of Mocamoon.
Okay, thank you.
Thank you.
Hi.
Hi.
So also thanks for the nice presentation. I have a rather maybe mean and critical question. For the real world use cases with the breast cancer TCGA set, you were showing that at the end of the day, you only found one active module between disease and control, and it was linked to DNA replication. Now I wanted to ask, is Mogamon too strict? Because that's very little information for a very big phenotype.
Yeah, well, the thing is that I was also shocked when I found this result, and I was even frustrated because I couldn't find any other active module. But when I looked at these Pareto front plots, this single active module was too far from the rest. So in terms of the optimization in the multi-objective genetic algorithm, there was no other module that could reach the same density or the same average node score. And this is why I decided to change the question, because it was the strongest signal between that would differ between patients and controls. But I don't think that it's too strict. It's just, I think it depends on the data set, because it's not that I control that, is that when it finds, let's say, even like a very, like a too good active module, then it would be very hard that another subnetwork will have the same or better density or better average node score.
Yeah, but you don't weigh the average node score or the density individually, or you evaluate the same way. Because I saw for your experiment plots that there were some subnetworks that had very high density but very low node score, and some other subnetworks had vice versa. So there was kind of a separation. So you didn't ever introduce a weighing parameter to kind of adjust for stuff like this.
Let me see if I can share my screen. Can you see my screen?
No, sorry, no. Oh, no, it's coming. Oh,
yes?
Yeah, now we can see.
Okay, so let me just find, here. So, yes, what you are saying, sorry, it's charging. Yeah, here. So, where is my pointer? Can you see my pointer?
Yes.
Okay. So yes, you are right. There will be cases. So with the multi-objective optimization, you will have cases in which the average node score will be very high. That means that you will have a lot of significantly differentially expressed genes, but the density will be very low. And you will have the other way around. You will have some modules that will have a very low average node score, which means too few or very few significantly differentially expressed genes, but a high density. But this is the advantage of a multi-objective genetic algorithm, because the two objectives are analyzed simultaneously. It means that you will have a trade-off between the two objectives.
Okay, so it's intentionally that you don't start weighing them individually. Okay.
Yes.
Perfect. Thanks a lot.
Thank you.
We have other questions in the chat. One question or remark, maybe. Great talk. Thanks, from Johan Dreo, probably. The results, he says, great talk. Thanks. The results part of the bioobjective algorithm make me think that you may find useful to consider the distribution of the Pareto fronts instead of samples of it. And there's an R package that he points to in the chat. I'm not sure quite that I understand the question, but maybe you can comment still.
I will have a look at this paper, but I really don't get why I would need the distribution of the Pareto fronts, because I actually take all the results into account. So when I run the algorithm 30 times, and this is very well explained in the paper, you can read it if you are interested in further details. But when we run the algorithm 30 times, at the end you end up with 30 different Pareto fronts, right? And then I take all the subnetworks that are in these 30 Pareto fronts, so these 30 first Pareto fronts, and then I rerun them again, and I just take the the ones that are best, so the ones that end up in the first Pareto front out of all the first Pareto fronts.
Yeah. Yeah, well, Johan, you have an opportunity to log on or you know, ask the question live if you want to continue the dialogue.
But I will read the paper. Thank you very much for the suggestion, Johan.
All right. Another comment from Farzaneh Firuzbakht. There's she says, thanks for the nice presentation. Your method involves multi-objective optimization of average node score and density. Could you please mention how you choose the best answer on the Pareto front? Okay, I think you're choosing all of them.
Yes, exactly. I do not choose only one, and this is the main difference between a classical mono-objective genetic algorithm and a multi-objective one, because in the multi-objective version, you have a set of solutions at the end to choose from.
She continues, yes.
Yes.
I think it's clear. The other question that she asks is how the Mogamon method switches between layers, and I had kind of a similar question, namely, you know, how you, whether there's a methodology, whether the Mogamon method does something extra to deal with multiple layers, or whether you're just building one network from multiple layers by connecting them, and then just run the method as you've described it before.
The second part of what you described would be to aggregate all the networks and to put them all together.
Yeah.
And we are avoiding this because if we do this, then it would mean that the densest network, which is the co-expression one, would just like run all the search. It will hide the other signals. So here I really have a multi-layer network, a multiplex, and the way that I change between layers is because every node is connected to itself across the layers. So every time, for instance, when I am doing, for example, when I'm creating a new individual, I start in a node, and then every time I want to add a new node, I ask, do you want to stay on the same layer or do you want to change? And this is a random process. So if it chooses to change, then it will change to the same node but in a different layer, and then it will have access to the second layer.
And then I'm assuming when you talk about genes and transcripts and proteins, it's clear how you identify nodes in different layers. But what do you do if you have microRNA, for instance, or other types, metabolites? Is that obvious how to do that?
Well, you will have, so in this case, we only had, as you said, genes which are associated to protein genes. But if you have microRNAs, I think you would just have to maybe add another, let me think, maybe add another layer that's,
there's some work for the future there, it sounds like.
Yes, I think so. Thank you.
All right. Do you have other questions in the chat or in the room or online? If that's not the case, thanks a lot again for having gone through the effort of recording your talk and joining us online under difficult circumstances.
Yes, thank you very much for the invitation.
All right, and with that, there's one more question I hear. Do you read it? I can read it on the chat, I guess. There's a follow-up comment from Johan Dreo. The distribution of Pareto fronts will be useful for assessing the efficiency of the search algorithm against other options, for instance. As you said, it would not be useful to extract the very best solution that you consider in the aftermath. I see. So he's talking about the efficiency of the search algorithm.
Okay, like maybe he means that it would be a tool or a way to measure if the distribution is like uniform. Like if you really find solutions that are, yeah, I think I understand what he means. So I will read the paper and I will consider it for further work.
All right.
Thank you so much. And then there is Crystal that asks, what exactly does the node score represent and how is it defined? The node score represents, the node score is the Z score that we normalized. So it represents the level of the regulation of the gene, whether it is upregulated or downregulated. So that's what it represents.
So it's when you compare, for instance, your cases and controls, and right? Or not?
Yes. Yes.
Yeah, okay. Yeah. Okay. Thanks for clarifying.
Thank you.
All right. I'm not seeing any other questions at this point. Thanks again, Elva. Have a good flight, and and we'll see everybody else, the online audience and people in this room, at 11 for the talk by Nataša Pržulj, which will also be on network analysis.
Yeah, thank you very much. If you have further suggestions, comments, or questions, do not hesitate in sending me an email. Thank you very much. Have a nice day.
Thank you. Bye-bye.
Bye.
Bye, everybody. Yes, see you at 11. Great. Welcome back to the second session of this morning. We're going to stay in the area of networks and network-based analysis for precision medicine. In the previous talk, we've heard a little bit about a paradigm that allows us to analyze networks, which is to take some data like transcriptomic data to project it on top and to integrate these two types of data. And our next speaker, who is Nataša Pržulj from the Barcelona Super Computing Centre, is somebody who has a lot of experience with this. She's pioneered the analysis of signaling networks and developed concepts that allow us to, other concepts than the one that we saw this morning, to analyze graphs and networks and the associated data. I'm really happy that she agreed to talk about these things and maybe future things that come up. We're all very curious, and you have the floor, Nataša.
Thank you very much. Thanks a lot. It's a pleasure, always pleasure to speak. I do have 1 hour, right, or so? Or how about the time? Or a bit more? I'm not sure.
You have one. Yes.
1 hour. Okay, good. And I will.
We have even a little bit more. We scheduled for you for 1.5 hours. So if you go a little bit longer, there's no problem. If you're shorter, that's also not a problem.
Okay, great. Thanks so much. Now to share, should I click this open share tray, right? To share the slide.
To share it next to the red button, I believe, in Teams on the upper right-hand side. There's a red button that don't leave with that, but the button next to it is a little rectangle with an arrow inside.
Yes, exactly.
Like that, you get to choose the window or the screen that you want to share.
Yes, I think I got it. Do you guys see my slides now?
We do see your slides, not yet in full screen mode, but
I am trying. Okay, now?
It's perfect that way.
Okay, thank you so much. Okay, thanks again for inviting me. It's a real pleasure to talk to so many people, young people included in this wonderful training network that I'm very proud to be a part of. So today I will talk about some of the new methodologies that we have been developing in my lab over the past 15 years or so. And it's going to do with the analysis, with the new algorithm dealing with the analysis of network data, how to fuse different kinds of network data to see what they collectively tell us, and maybe how to formulate new paradigms from the data, bottom up, in a bottom-up way. And this is an overview of my today's talk. So basically we see biology and medicine in general as a complex world of interconnected entities that need to be mined to uncover new information that's currently hidden from us in the complexity of these data. The talk will be structured as follows. First, I'll do some motivation, maybe even some philosophy to know why we are doing what we are doing, where we are headed, and why this is needed right now in this moment in the history of science. And then I will overview some of these new methods. I will only have the time to go through, through some examples of the new methods that we have developed to mine these large-scale, multi-scale interconnected data. And The new methods will be in two different classes. How do we analyze single type of omic data? For instance, one type of a molecular network, either presented as pairwise binary interactions between the entities, or as multi-scale networks of not only pairwise interactions, but more than pairwise, where multiple entities can interact simultaneously, and how by modeling and mining the data from these modeling formalisms, we can learn more about biological function or disease. I'll present some diseases. For instance, I'll present some of our work on cancer, leukemia, and maybe some rare disease. And then the second kind of methods that builds upon these first kinds of methods is how to analyze multiple layers of heterogeneous network data simultaneously. And I will present our paper from Nature Communications from a couple of years ago, where we made this new paradigm, new model of an integrated cell that we call an I cell, how we applied it to several kinds of cancers. Also, I will present how we utilize this framework and actually adopted to do patient-centered data integration to improve multiple tasks of precision medicine at the same time and at the same computation costs, such as stratification, better certification of patients of the same disease, how we uncover new biomarkers, how can we repurpose drugs. And here I will also talk about COVID-19, how we applied it to COVID-19 and what we have learned from the new COVID-19 data. I will only briefly mention some other works in the area, basically how from bottom-up mining collectively of these data, we learned about other things such as reclassifying diseases, reconstructing gene ontology, network alignment, etc. And in the end, I will conclude again about perhaps, well, some, of course, technical challenges that we are facing, but also some of the philosophical issues where we are headed. And I will only have the time to give a pretty much high-level overview, and I will not go into, I will illustrate how the methods work, but the excruciating technical details you can read from the papers, and everything I talk about, they will be underneath a reference to the paper where you can read the full detail. I thought this would be a better use of my time rather than having a tutorial of the technical details. So let's begin with the motivation. We've witnessed over the past decade and a half or a couple of decades that technological advances in experimental biologies have yielded an astounding harvest of various molecular and clinical data, from the genomes and now more recently epigenomes to transcriptomes, proteomes, metabolomes, metagenomes, phenomes, exposomes, etc. We've been amassing these data at unprecedented rates ever seen in the history. This data growth was guided by empirical reductionism that was striving to dissect the biological entity into its constituent parts to better understand it. However, even Charles Darwin, in his Origin of Species, wrote that a biology is a tangled bank, as he said, with all of its aspects interconnected. At the same time, concurrently with Darwin, Virchow's pioneering observation in Germany that all diseases involve changes in normal cells has forever changed the way we practice medicine. So we know that knowing parts is not enough. And right now, this data growth about a cell has made us hit a wall of biocomplexity. We know that cells are not just loosely coupled arrangements of quasi-independent molecules, but highly intricately and precisely integrated networks of entities and interactions within the cell and with the environment. All of these data types complement each other. So basically, each biotechnology that produces these data, whether it produces protein interactions or expression or whatever, these are just like different pair of glasses that we put on to look at the same thing, at the cell. And very likely into the human cell, because this is what we care the most about. And this is why all of these data types, they're just different slices, different things that we measure in the cell, this is why we have to all model and mine them within the same framework, because each one of them contains only partial information. So I believe that the time has come to replace the mostly reductionist molecular perspective that dominated the 20th century with a new and a holistic view of the living world that's required to explain biological and medical phenomena and biology's innate complexity. However, this requires establishing a perspective and framework not only for one problem, but for biology and medicine in general. With the foremost challenge that we are facing now is how to re-synthesize biology, how to put the elements, all of the elements, back into their complex, dynamic environments, how to connect them all within a unified framework, and how to reformulate biological paradigms within our non-linear world. And this requires a vision, and we are hoping to bridge this gap by developing mathematically principled frameworks for integration of all of these interconnected data. And to do that, we have to marry biomedical problems and data with the algorithms from all sorts of computational sciences, such as machine learning, mathematical nonlinear optimization, network science, algebraic topology, etc. And even when we do that, we need high performance computing to solve our algorithms, to run them on the data, because our algorithms fall, our problems fall into the category of so-called computationally intractable problems. So pretty much we can only do heuristic algorithms, hoping that they work well for our data only. And in general, of course, they will fail, because this is what computational complexity tells us. This comes from the theory of computation. So you will see in this talk how we propose modeling and computational advances that we hope will link the medicine's reductionist past with its holistic future and enable displacement of the dominant molecular representation of biology that has yielded this large quantity of data that I just talked about, by a new and integrative paradigm that is deeper, more comprehensive, and more inspiring. And I currently have an ERC consolidator grant to do these kinds of things. So these are the results of that grant. Also, we are trying to commercialize these results, which is actually not trivial. The business world is not very different from the academic world, but I've seen that within this program, you're getting some training on that, which is quite nice. And also, I will present some of the results obtained during my ERC starting grant that preceded the consolidator grant, and it enabled me to come to Europe from the US. Now, the computational challenges that I talked about just a few moments ago are the following. So, for instance, let's take a look at the genetic sequences. We've had these sequences for quite some time now. And even though the problems of aligning, for instance, or comparing these linear objects, these sequences, these problems fall into the category of so-called computationally easy problems, meaning that in the time polynomial in the size of the input data, you can get the exact result. And even though we don't have a very good understanding of what the genome does. Now, dealing with large network data is much more complicated. For instance, just aligning or comparing large networks is computationally intractable, computationally hard. And Stephen Cook, a professor from the University of Toronto, has proven this in 1969 in his seminal work, basically, where he made the foundations of computational complexity. Basically, in that work, basically, it was done at UC Berkeley, and then because he didn't get tenure there, he moved to U of T, where basically he worked all his life till retirement. And in these seminal works, he started a whole branch of computer science, complexity theory, where he has shown that some problems we can solve, while other problems we cannot solve. Okay, so these are so-called computationally intractable problems and various classes. So one of these problems is so-called subgraph isomorphism problems. In a nutshell, asking whether one graph exists as a copy in another graph. And on large data, you can mathematically prove that you cannot solve this problem exactly, even given all the time of the universe and all the compute power of the universe. It's impossible. At least this is currently believed, unless somebody proves $1 million question whether people, but let's, this is the theory of computation. So even comparing large networks is computationally intractable, meaning that the only way we can solve this is by making approximate algorithms, to approximately solve them. These algorithms are called heuristics, and because of computational intractability, these algorithms have to be carefully tuned to your particular data, because on another problem they will fail, and this is what computational interactive actually means. So this is why we have computational challenges to extract information out of each of these individual layers of information about the cell, whether it's the networks of genes and their co-expressions under particular conditions, or whether it's physical bindings between proteins and protein-protein interaction networks, or whether it's epistatic genetic interactions, so double mutants, where a single mutant produces a certain phenotype, but double mutant produces a phenotype that's not expected from the behavior of the single mutants. Also, we have metabolic interactions. We have drugs that we can connect in a network. For instance, if they have a similar chemical, if they're chemically similar, they have high chemical similarities, or if they cause the same side effects, or if they can be given to the same patient, or if they can be prescribed for the same disease, etc. So you can make various kinds of networks on drugs. Then you have ontologies such as disease ontology and gene ontology that I talked about. So each one of these, first, to analyze them, to learn what they tell us, we have to make these heuristic approximate algorithms to extract the meaning, the new information out of them, the new knowledge out of them. And then also, even when we do that, how to mine them collectively. And even if we can do that, how to put that in a patient-centric way. So these are the great computational challenges, pretty much all computation intractable. And with this motivation, I would end the motivation and start talking about these examples of how to mine one type of omic data, let's say molecular network. And network that we did a lot of work on is the network of protein-protein interactions. So basically, just to extract a little bit of complexity of these molecules of proteins, we model them as nodes in these networks. So each node corresponds to a protein, and then if physical binding is possible, then we put an edge between the nodes. And then, given a healthy and a disease network, we want to compare them. And we just saw that this is computationally intractable. And this is exactly why people have been doing some simple approximate ways how to tell whether two networks are similar or not. So they would compare, for instance, their sizes, the number of nodes and edges or links, or the degree, so-called degree distribution. So the degree of a node is the number of links it has to other nodes. Okay? And then you take over all nodes, the distribution of these links, or so-called degrees, and this is your degree distribution. So, to approximately tell whether two networks are of similar structure, you compare their degree distributions. But then very quickly you can come up with these examples, and this is just a simple illustration why and how every heuristic fails. So for instance, here you have two networks, G and H, that are of exactly the same size, meaning they have exactly the same number of nodes and exactly the same number of links. They have exactly the same degree distribution. Each node in each of these networks has degree 2. It's linked to 2 other nodes. And yet you can just look at them and see how very different they are. They have very different behaviors. So for instance, H is connected, G is disconnected, etc. So this is why quite a while ago now, in 2004, I came up with so-called graphlets. These are small graphs because another word for a network is a graph. And I came up with these small ones, all of them, with a small number of nodes. And I view them as Legos of networks. And I ask whether by the way they're put together, kind of in a Lego terms, in the end you have a pumpkin or you have an airplane. And this was a subject of my starting grant. But then rather than just counting how many of these guys appear in your data, at the same computational cost, you can do much more by noticing these symmetries with it. While an edge was symmetric, it didn't matter where a node touched an edge. It matters where a node touches a three-node path, because three-node path is not symmetric. The end nodes look the same, but they look different from the middle node. Triangle, all nodes look the same. It's a completely symmetric, but then four-node path is not. End nodes look the same, but different from the middle nodes, etc. And these are so-called automorphism orbits. Again, this is technical detail that we can go into detail later on, or you can just read in this paper that I introduced them. But let me just illustrate how that works. Let's say for this node A in this small network here, for each of these orbits, okay, so for each of these symmetry groups, for each of these graphlets with up to 4 nodes, I will count how many times node A touches each one of them at that particular orbit, at that particular location. So for instance, node A touches 1, 2, 3, 4 edges. This is why this coordinate is 4. Then let's see how many three-node paths it touches at the end. 1, 2, 3. It touches 3 three-node paths at an end. How many of them it touches in the middle? Then you need to start counting how many of them it touches in the middle. This one, 1, 2, 3, 4. Maybe there is another one that I'm missing, probably. And this is how you count. How many triangles it touches? 1 triangle. This is why this coordinate is 1. How many squares it touches? 1. This is why this coordinate corresponding to a square is 1. So basically now for each node in the network, you count very precisely its local neighborhood. And then you do this for all nodes in the network. And then you have this matrix. And then you can go along the rows and compare these vectors, these basically feature vectors, measuring the structure, the topology around the node, to see how similar the wiring around the nodes are. Or you can go along the columns. If you go along this first column, or 0th column, this will be your degree distribution, measuring basically the distribution of over all nodes of the degrees of the nodes. Yeah? Or this would be orbit 1 distribution, orbit 2 distribution, orbit 3 distributions. And very quickly, you have very many distributions by which you're measuring the topology of the networks, and then 2 networks you can compare in each of these distributions. So in this, and then you can do many other fancy things with these matrices. This is just an illustration, and we've done a lot. Now, as I mentioned in the motivation, network alignment is an intractable problem. It is still an active area of research. So just as we want to align or compare genomes or sequences, genetic sequences, genomic sequences, which has revolutionized our understanding of biology, we want to do the same with the networks. But this is much harder, computationally tractable. And this is why over the years we invented this family of so-called graph aligners or graphs. The last one we published like 4 years ago, unified alignment. You can see how that's done. Some of these are based on seed and extend kinds of approaches, others on various optimization things, etc. Now, just to briefly mention that very early on, we started applying these methodologies to biology. So, for instance, even in 2008, we've shown that similarity in the wiring in terms of these graphlets around nodes in the PI network means the similarity in biological function, membership in the same protein complexes, subcellular localization, the same tissue co-expression in human, and the same disease involvement. And of course, for the nodes that are in the similar, in the neighborhood of a PPI network, protein-protein fraction network, but also the nodes that are far away in the network. And not only the similarity in the wiring patterns, even in far away neighbors, means this biological, functional similarity, but also it means a similarity in the involvement of disease. And that carries across life on Earth. For instance, here we have a simple machine learning method based on canonical correlation analysis, where we correlate this linear combination of these graphlet degrees with the linear combination of gene ontology annotations. And we see that, for instance, even in species as diverse as yeast and human, membrane proteins look like these hubby ones, while transcription factors are more cliquish. And as I mentioned very early on, we applied it to cancer research, and we got phenotypic validations of our observations by siRNA screens in Professor Gaddison's lab at UC Irvine Dermatology Department. We found that just by this topological similarity in protein interaction network, and even in these data that were very noisy back at the time, now we have much better quality of these data, but these measures are very robust to noise. So we identify new members of melanin production pathways, this is important to find these guys, because then maybe they can be targets of drugs. Now, coming to more recent work from last year that we published at ISMB, this is a work now on a different kind of network data. This is high-C data on chromatin structure. And we apply this one now not to cancer, but to CLL kind of leukemia. And we analyzed the CLL, leukemia cells, and the control cells of origin, B cells. And this is an illustration of these chromatin structure network data when we did the visualization of its modularity or clustering structure. You see that the control cell, the proxy for a healthy cell, you clearly see the modular organization. While the CLL chromatin structure network, you clearly see disruption of any kind of organization. And biologists will say, oh, well, we knew this. I mean, of course, cancer disrupts the organization. But what was not known previously was, and this is the first time we discovered it, that this structural difference of the driver genes exists even before CLL hits. So the structural difference, basically where cancer hits, you know, the specific points in the chromatin where it is present. And in this way, by just identifying these, then you can find new disease-involved genes, new genes important for CLL. You can read more in that paper about the details, but basically just application of replication of the previous published methodological work. Now, as I briefly mentioned, this methodology is very robust to noise, depending on the kind of algorithm, but even in the presence of up to 70% of noise, of random noise, we can still detect signal. It is the best performing on the market right now. We have generalized it to directed networks. We can track the dynamics of the networks, how the networks change over time, and how particular nodes change over time in these data. Also, very interestingly, in 2004, in the same paper where I invented graphlets, proposed them for the first time, I've proposed that protein-protein interaction networks are geometric, that they have geometric structure. That has been validated later on, not just by our lab, but by the labs of others. And you see now, it's 10 years, the so-called topological data analysis developed. Also, you've seen, I don't know, machine learning kinds of methods employing geometricity, etc. Also recently, also last year, now at ECCB conference, European Conference on Computational Biology, we generalized these graphlets to work with probabilities on the edges. And we've shown that if you include probabilities on the edges, that you can better identify condition-specific functions. So you can read about that in that paper. Also, a couple of years ago, we generalized Laplacians, just normal Laplacians, to so-called graphled Laplacians to capture not only the direct neighborhoods, like the normal Laplacian does, but also the graphlet-based neighborhoods, so extended neighborhoods. And then based on these, we also generalized spectral embedding and clustering to work with graphlet Laplacians. And we've demonstrated that we can identify different and complementary annotations by including these extended neighborhoods. So just another methodology that demonstrates the same. Now, graphlets as Legos of networks have not only been used in biology or medicine, they've been in other areas, for instance, in image categories recognition, photocropping, for scene classifications, etc. Also in social networks for face recognition, for instance, in large organizations to maximize productivity. Also in theoretical computer science, etc. Now, before I start talking about this data integration or data fusion, let me just mention extension of these graphlets, how we can model and mine multi-scale organization of the data. So in the top figure, you see that one protein complex with 3 proteins, we model by 3 nodes corresponding to proteins and 3 links, 3 edges, 3 interactions between them. But also if you have 3 protein complexes containing 2 proteins each, you also model in the same way. And this is basically where by modeling you blur the information. You have to, you have the same model for 2 different things. While actually a better way to capture this is to model the left-hand side protein complex by 1 set of 3 nodes, and to model the phenomenon on the right-hand side by 3 sets of 2 nodes each. And this is exactly captured by hypergraphs. Hypergraphs is an extension of graphs, where you have nodes just like in normal graphs, but an edge is not defined only between 2 vertices, between 2 nodes, but between any set. So any set of nodes can be an edge. So there's a so-called hyper edges. They are sets of many nodes, or it can be 1. It doesn't matter. So this is an old notion in graph theory. Claude Berge published a book on the theory of them in 1989. But these notions are difficult to work with. And before our work in systems biology, they were not used much. Ernesto Estrada, who was in England, but now he moved to Zaragoza, I believe, defined only the centrality and clustering measures. In these terms of hypergraphs, degree distribution was in 2008 defined, and also just some general modeling considerations were made. And then also at ECCB 3 years ago, my former PhD student, who now graduated and is a director in one of the startups in London, he proposed the following model. So to generalize hypergraphlets, first to measure, to model both binary pairwise interactions between proteins, but also protein complexes and pathways as hyper edges. This is a so-called PIH model of multi-scale protein interactions. Then a hyper, define hypergraphlets and also hypergraphlet orbits. And in this way, you actually are dealing now with combinatorial explosion. This is why you don't need high in these counting these even graphlets. But up to 3-node graphlets, we see that we have 64 now, feature vector of 64, dimension 64. And then if you go to 4-node hypergraphlets, you have over 6,000 features in these vectors, right? And then basically then again, you compare these feature vectors, so-called hypergraphic degree vectors, cluster the data based on them, and then again you can use canonical correlation analysis to link between, let's say, gene ontology, biological process annotations, and these orbit counts. And you see that similar functions, for instance, these kind of learning, memory, and neuronal synaptic functions all look like this. They're characterized by these kinds of orbits. We're not too sure why, but this is the signal we're getting from the data. And also you can read the details in this paper, of course, but by modeling and mining the data using these formalisms, you can get much higher functional enrichments, meaning that you capture more biological information and then you can exploit that to learn more about biology. Also, last year, with colleagues from America, Peter Radivojats and his former PhD student, who is now, I believe, a postdoc at MIT, we worked on hypergraphed kernels and how to classify biological networks with them. You can read about that in this paper that appeared about 1 year ago. Similarly, 2 years ago, we played with a different but similar notion of, we generalized graphlets into so-called simplets. This is similar to hypergraphlets, but still a different notion, because now we are generalizing, basically we're modeling the data and using abstract sequential complexes. What are these? This is basically a family of sets that is closed undertaking subsets. So in that sense, hypergraphs is a more giving notion. It's more flexible, but nevertheless, we model the data by using this. So a protein interaction network, a PPI network, is a one-dimensional simplex, but then we took also the protein complexes. So we made this new model, and we modeled human and yeast protein interaction networks by this. We call this PPI complex model. And then when we generalize graphlets to these simplets that we saw on the previous page, and do exactly the same, like feature vectors, do the same clustering and enrichment analysis, we see that with this PPI complex representation and with simplets, we capture more biological information. We get higher enrichment than just dealing with pairwise interactions. And this random is here at 0, so this is why you see. Okay, so I would close this section here because I would like to spend some time on telling you about the multiple layers of heterogeneous data. And the reason why I talked about these networks is because we use them, we built upon them, we use them in our further, we built upon them in our further methodology of fusion, data fusion of different but complementary data types. Excuse me. So, in this paper, I'll just illustrate this for this paper. First, from Nature Communications from 2 years ago, we modeled cancer and the healthy or control, let's say proxy for healthy cells of various kinds of cancer. And we took the following kinds of data. We took gene co-expressions in a particular cancer, so we made a network. We took protein-protein interactions from I2D network. I think now it's out of service, but these data are available everywhere now. Then we also took genetic interactions from BioGrid and the synthetic lethality database. So these different kinds of networks. And we also took tissue-specific gene expressions from the Human Protein Atlas. So we made these tissue-specific networks for a particular cancer and a particular controlled cell. We made this methodology to fuse them. You know, I briefly mentioned it last week, but then when I talked for 10, 15 minutes, and also Katarina talked about these kinds of methods, but maybe let's give a bit more detail here or not. I don't know. And then once we have this integrated cell, this is different network. This is our model of cell. Then we compare them using these graphlets. This is why graphlets are important, and we find new cancer genes that we phenotypically validate in our lab. Now, why do we want this new data fusion methodology? When we took these data from the databases that I just showed you, protein interactions, co-expressions, and genetic interactions, we saw that in the number of genes, there is a large overlap. There's around 3,000 genes that they have in common, but actually that they have one shared interaction. One. And this is not a fiction. I mean, this is real. Basically, you're just measuring different things by different methodologies. And this is why each of these slices of the information just gives you partial information. And actually, because of this low overlap, or actually we can say no overlap, This is why other methodologies, competing methodologies, failed. And at the time, 2018-19, we tested all of them that were on the market: Markov clustering, graph use, spectral clustering of multi-layer graphs, invariant network fusion, and they fail. And it's because of this empty overlap. They either return an empty integrated network, or they diverge after a small number of iterations, or they have memory issues. They can improve, or they don't produce clusters, or they produce but very large number of very small clusters. We leave many nodes isolated, etc. Why we needed to make a new method. So we created the tissue-specific networks from these particular data sources, as I mentioned. Now, notice that when I show you the results, these tissue-specific gene expressions are from the Human Protein Atlas. They are not microarray or RNA-seq or anything. It's antibody staining. They're low throughput, low number of samples. They don't represent all patients. There's a small number of patients. So if you don't see a favorite gene in there, it's because it's not, there's something wrong with the method. It was not in the data. This is not high throughput data. And we only consider genes with expression values available in the Human Protein Atlas, and if they have more than one protein-protein interaction. So we created the tissue-specific networks where nodes are genes that are expressed in the tissues as per the Human Protein Atlas, and they're linked by the interactions from here. Okay? So for now, let's focus on 4 most prevalent cancers in human: breast, prostate, lung, and colorectal. These are the control cells of origin. And then these network statistics, some people like to see this. The size is the number of nodes and edges in each of these kinds of data from these databases: protein interactions, co-expression networks, and genetic interactions. And you can see here in red that you have on the order of 10,000 nodes and 100,000 interactions. Perhaps for genetic interactions, a little bit less. But you have a lot of data, quite enough to analyze by network statistics. This is the methodology that Katarina was talking about in her talks. So now we have these 3 kinds of networks: PPI, co-expression, and genetic interactions. We represent them in a standard way in a computer by matrices. You know, this is genes by genes, and if there is an interaction, there is a 1 here, or if there is no interaction, there is 0. Okay, and then in co-expressions, you have something similar. You can have the real value. You don't have to have 0 or 1, and genetic interactions similar. So these 3 matrices now in the computer, we are approximating by the 3 products of matrices: G times S times G transpose, G times S2 times G transpose, G times S3 times G transpose. Okay, so, and we do this, now this is a continuous optimization problem, also computationally intractable. We do solve it by minimizing this objective function on large data, of course, like we have. So this G contains, this is the genes, this is all the genes that we have, and this is low dimensional here. And this low dimensionality is basically our parameter to tune. But after you resolve all that, you can read the details in the supplement of this paper. Then we multiply G times G transpose to get another network, genes times genes, that we call an integrated cell or an I cell. And then we find functionally coherent domains in it. Not only we find functionally coherent domains, but we find that the I cell is more. So this new network, this new model of molecular organization of the cell is more functionally coherent than its networks of origin, PPI, coax, and GI, in the sense that you get higher functional enrichments in many different kinds of biological annotations. This particular one is by a reactant pathway annotation. So basically, when you do the clustering, this is actually a kind of a co-clustering, and this is equivalent to kinase, etc. And this whole thing about factorizations, whether it's just a factorization or 3 factors, which there's 2 factors or 3 factors, has been around for about a decade, more than that. Okay, but the applications of it in this area are fairly recent, only about maybe 7 years old or so. Now, these also I cells are non-random. Now, this is a whole other field of families of graphs. So there are so-called Shreni random graphs, there are other random graphs, network random graphs. Structured families of graphs is an area on itself. I did my master's in that area. Half of my PhD was in the University of graphs. But let's leave that for now. Usually biologists are not concerned about this, but the importance of these structured families of graphs is in the following. If you can uncover the structure, which family of graphs, which family of networks the data belongs, then we know usually the theory of that specific kind, family of graphs or networks, then you can make fast heuristic algorithms. This is the purpose of this whole thing, and this is why I proposed geometric graphs in the first place. Now, whatever, 16, 17 years ago. As we saw, PPI or co-expression or genetic interactions are well modeled by some families. iCell is not. We don't know which family of graphs it belongs to, and this is something we have not done much yet. But here also, let me just show you that the contribution of, basically each data type contributes to the quality, to the biological quality. And this is the biological quality of the clusters, of the clusters, co-clustering technique, right? Measured by KEGG pathways, reactome pathways, and gene ontology, biological process annotations. And you can see that, for instance, you can forget about these two, A2 and A3. You can only decompose one network, only protein interactions. And this is how much enrichment you will get in the resulting clusters, or if you can only decompose co-expression or genetic interaction network. But when you start integrating more data into the model, you can see that basically when you put all data in, you get very high quality, much higher than from each of the constituent data types in isolation. Okay, so now we created tissue-specific networks, protein interaction, genetic interaction, co-expression networks, and also iCell networks of these 4 different kinds of cancers and control, tissues of origin. And then we went again into the human protein atlas, where a gene is either expressed or not in the tissue, or maybe it's differentially expressed, but let me come to that later. And then we find between cancer and control tissues the genes that are cancer silenced, cancer activated, always silenced or not expressed in either cancer or control, and always expressed both in cancer and control, maybe at different levels. That's what I'm saying, maybe differentially expressed. However, we find that cancer silencing and activation is not that relevant, but actually that it is these always expressed genes that are enriched in drivers. And this is what made us to study these always expressed genes in more detail and their involvement in cancer to maybe predict new ones, and especially the wiring of them. And then we took cancer and control, iCells, PPI, GI, and coax networks. And we found the most rewired, this is GDV rewired, meaning the most rewired that we measured by graphlets. So the most rewired in cancer versus control. And check their enrichment in drive. And only in iCells, not in PPI, not in GI, not in co-expression, not in the data, but only in the model, only in the iCell model, we found that rewiring is independent. When we found that out, we took the top 20 of the most rewired genes in cancer iCells of these 4 cancers that I talked about. So there would be 80 of them, but 63 would be unique because some of them repeated. And we validated them almost all, either through literature curation or by our own knockdown experiments. We would knock down each one of these and measure cell viability. And then we found that, basically, if you take this cancer cell line and you knock down these particular genes, then you get significantly reduced cell viability in cancer, right? Or maybe here in this colon cancer cell line, significantly increased cell viability in cancer. So these are the kinds of experiments we validated them. And also, as the third kind of validation, we did retrospective analysis of patient survivals. And we found significant differences in the survival of Okay. Now let's get to this differential expression. We found out that only 17 out of these 63 genes, or 27%, are differentially expressed in cancer. So not even through differential expression you can get to this. Once we figured this out, we went pan-cancer. We took 16 more cancers from TCGA, so 20 cancers in total. They are all listed here. We made their cancer eye cells because we figured out eye cell is important. And then we found similarly wired genes in all of these different cancer eye cells and asked if they are cancer related. We found all of them that are always expressed and cancer expressed, around 3,000 of them. And we found the most wiring similar of them, common to all cancer eye cells. And we found that they are enriched, significantly enriched in drivers. And then we validated the top 20 of them, either through literature curation, or we found them through some databases that we did not use in this study as the data to compute on. But actually our top predictor was nowhere to be found as involved, this is mitochondrial, Nadix hydrolase. And we found no evidence anywhere that it's involved in cancer, but then we went to the Human Protein Atlas and we found that significant differences in patient survival with patients of these mutations in these genes in 8 different kinds of cancer. So, to conclude, we have a new concept of an integrated cell. This is a new model, a molecular organization of a cell that integrates different kinds of data. In this case, 3 particular kinds. You can put any others if you want. And we did specific cancer-specific and pan-cancer studies. We've shown that all data complement each other. They contribute to the quality of our model, and that, but they all have only partial information, including differential expression, and we need to go beyond. This is a generic framework, and basically now I will show you, this was the first example on cancer, but let me show you what else we did. And this methodology enables integrated analysis various kinds of applications. For instance, this is an ongoing work, and actually it's not even ongoing, it's finished, and we revised and resubmitted this paper. Hopefully it will see the light of day soon. This is for rare disease. So in rare disease, as many of you know, the problem is a lack of data. You have very few families. And this is a rare form of thromophilia, so-called the Belgrade mutation that appears only in northern Italy, in the Balkans, and in the Far East. And we only had 2 families, 2 brothers, one diseased, one healthy, but the healthy brother had diseased daughter. And we have 2 sisters with the disease. And because with so few patients, you don't have statistical power to analyze the genomes, then by doing these sorts of integrative analysis, incorporating more data, you boost the signal. And here we have the genes and different kinds of networks on the genes, like we saw in the eye cell that I just talked about. We have protein interactions, co-expressions, genetic interactions. But then we also have germline mutations. We have this matrix going from genes to these patients. Here we have only 5 patients, but nevertheless. We posed this model, we solved this model, and we, for instance, one of the results, just to highlight, we found this cluster of connective cluster in the molecular interaction network characterized by a lot of mutated genes, but only in diseased women and in all diseased women, while this cluster, another connected cluster, contained the mutated gene that are mutated only in the healthy brother. So our hypothesis is that actually this brother was lucky to have these mutations that protect him from the disease, but he passed the disease on. This is to be validated. For now, it's just a computational study, but of real data of these real patients. Now, let me show you other examples. As I told you how versatile the methodology is. This is a machine learning methodology. It enables you to make predictions, not just to do co-clustering. While in the eye cell, just as I showed you in the previous example, we only worked on genes, and we had different kinds of interaction networks on genes only. Nothing prevents you to have different kinds, different types of data between different types of entities. For instance, we have patients, and then we have, let's say, somatic mutation profiles between patients and the genes. I have not just 5 patients like we had before, but we have hundreds, thousands tens of thousands of patients here. Then another data type, for instance, that you can have is drugs. So you can have, for instance, links between patients who have taken particular drugs or who could take particular drugs. You can have drug-target interactions modeled by this matrix here between drugs and genes as proxies of proteins. Then you can have various networks on the drugs, for instance, chemical similarities. You can have patients, for instance, and their profile similarities as a network, etc. Now, basically, you pose this system, and then you simultaneously can decompose all of these matrices across by solving this minimization function. You can constrain it by the structure of these networks, by the topology of the data. This is just an illustration of one study that we did, but we didn't publish, unfortunately, from London. These were patients. Basically, there were about 40-something patients, cancer patients, and you could see that a patient would typically have very small number of mutations, but there would be an occasional patient here who would have maybe 20 mutated genes. Similarly, these 40-ish patients in total had about 180-ish mutated genes. Usually a gene is mutated in one patient, but there would be, you know, this usual suspect like 50, 53 that would be mutated in half of the patients. So this matrix is actually this matrix here, D13, between patients and genes, for instance. Now, once you minimize this function, you decompose. So what enables the fusion of the data is these matrix factors that you share across. The red one corresponding to genes, the blue one corresponding to drugs, and the green one corresponding to clustering, but not only clustering. By this matrix completion property, you predict, you make new predictions. And the reason why we like this machine learning method is not only because it captures all of the data, systems level data, and how the data relate. So your data model, you know, does not suffer from any sort of information loss, like in the other methods, machine learning methods, but also it provides mechanistic explanations. For instance, if you want to repurpose this drug to this particular patient, you can go look up in the system and say, okay, this patient has mutations in these particular genes that this drug binds to, and this is why you should repurpose this drug to this. You know, you cannot just, basically, these are the kinds of things that medical practitioners ask you for before they are going to do anything with the patient. Now, this is just a theoretical model and some study that we didn't publish, but the one that we did publish does not have all of these components. It does have the data from TCGA. These are serous ovarian cancer patients, somatic mutation profiles, patients to genes. It does have drug-target interactions from drug bank. It does have drug chemical similarity network on drugs from drug bank also by SMILES. And it does have these three kinds of protein interaction networks from BioGrid and KEGG. And we decomposed these matrices across the different kinds of objects, the heterogeneous objects, into these three matrix factors. And then basically from this same, once this converged, from this same minimization function, from this same mathematical formula, we got all three things that we wanted to do. Patient stratification from this cluster of patients. We got cancer gene predictions from this cluster of genes that were significantly enriched in drivers, for instance, and drug repurposing by matrix completion property of this drug-target interaction. And you can read more from it in this paper for 5 years ago in the Pacific Symposium on biocomputing. Okay, now through fivefold cross-validation, you can see here again how if you only decompose drug-target interactions, only one kind of data, to reconstruct it, you're this much accurate in terms of the area under ROC curve or precision recall curve. But when you add all of the data, you know, as you keep adding data, you're better and better. When you add data, you're fairly precise with fairly small. Here I would end about this. You can read more details on biological details from the paper, because I would like to spend a few moments, and you've already been, well, not quite an hour, but maybe 50, 55 minutes. So give me maybe a few more minutes to talk how we utilize this in COVID. And we published this a couple of months ago in scientific reports. So Of course, like everybody else, like the entire scientific community, we were mesmerized by this huge problem that challenges our society. And very early on in the pandemic, in the early 2000s, we got data. Actually, the data, the patient data, and the viral host interaction data. So we took these data. The data on viral host interactions we took from this protein interaction map. So basically, these were the two key papers. One had the data on to which human proteins, viral proteins bind, and the other one had the data on differential expression, on basically differences in the expressions in the lungs of the infected patients after they infected with this disease. So these are the data that we took. As I said, molecular interaction networks, like you have seen before, drug-target interactions and drug-chemical similarity. These are the databases that we took the data from. We had the data of 26 viral proteins to around 330 human proteins. These are the bindings. We had molecular interaction network, as I said, drug-target interactions and drug-chemical similarities. And we posed this system very similar to before. This was the matrix that was shared across human genes, and this K2 is a low-dimensional factor. Then molecular interaction networks were and drug-chemical similarity networks were used as these constraints on the minimization. And basically, by co-clustering of genes and drugs, we got new information there. And then by matrix completion of this drug-target interaction matrix, we got the drugs to be replaced. So that's in a nutshell. And then about the methodology. So nothing much new about the methodology. And then just the highlights of the data from these clusters of genes. You see that, we get a high enrichment also in the clusters of drugs. You see that for matrix completion for drug repurposing, we have very high area under the log curve. So precision recall curve is here. We did some validation of predicted drug-target interactions. We focused to reduce our space only on FDA-approved drugs for now. We found around 370 drugs targeting around 150 human proteins. So the reason what we did here is, rather than looking for antivirals, which are very difficult to obtain, we said, look, there's so many drugs and they bind to so many human proteins. There must be something out there that can help the patient. So this is why we focus on the host, on the human now, rather than on the antivirals. And basically, we found that out of our, basically, we validated in external databases 87 of our newly predicted drug-target interactions. Very interestingly here, so this is a kind of an interesting thing that we found out. So we did this data fusion. And then we asked, why? I mean, what is going on? These drugs that we want to repurpose, where are they? What are their properties? And we found something very interesting. When we did these viral interactions, so basically human genes that interact with the viruses, this is not where it was predicted. So normally people would, hit with these receptors with the drugs. Yeah? This is not where we want to hit by this methodology. Or people will say, okay, differential express genes in lungs, make these genes the same, modulate them to be the same as in the normal tissue. No, this is not what we got. What we got is that in the molecular interaction networks, the neighbors, the common neighbors, so you go into the neighborhood of the receptors that interact with viral proteins and into the neighborhood of differential express genes. So where these neighborhoods intersect, this is where these drugs should hit. So in between. So this is the importance of this study. And then when we looked at these neighbors, so this is what we got with by data fusion, that this is where you should hit. We found that these common neighbors much higher degree, clustering coefficient, eigenvector centrality, between the centrality, etc. And this is statistically significant difference. You can read the details from the paper. Again, by graphlets, we got this statistically significant difference of these common neighbors. Of course, you can get these common neighbors, their functional enrichments, and you see that they're functionally enriched in these viral processes, while the rest, the other sets, such as background genes or differential expressed ones, no, they're not functions. And now we are extending this methodology further. And you can read about the details of the biology of these FDA-approved drugs. We found about 180 of them hitting, 150 of them hitting about 50 human genes. We validated 11% of these FDA-approved drugs. 11% of them are already in government trials, and additional 17% are in this corona drug interactions database already. The functional enrichment of these 50-ish human genes that we identified. By these methods, we found in various annotation databases. Interestingly, what links them all is the enrichments in the following pathways: in the VEGF pathway, vesicular-endothelial growth factor, and nitric oxide signaling. And basically, all of these other functions converge there. So we assume that this is something that should be modulated in COVID. But the drugs that we found are cancer drugs. I mean, this is really very toxic. You don't really want to give them to patients. In any case, we proposed some things, and then it's up to the scientific community to further take this into biology. And these are our top 10 drug-target interactions and their scores and the external databases where we validated them, etc. So I would like to conclude here. So we saw that by data fusion, we found new drug-target interactions and also this new set of genes that needs to be hit that are neither receptors nor differentially expressed genes, but the linkers, those in between, the common neighbors, and that are very topologically different, and in addition to that, that they're enriched in viral functions. So I would end talking about this methodology of ours and our work on COVID. We do have another thing, maybe it's already submitted, I'm not sure, or it's about to be submitted. Now, let me just mention that this methodology, we've seen this factorization methodology, factorization-based methodology, utilized in many different domains, and people ask, how can it work everywhere? But it's the same, like it's the methodology, like how can addition work both in a grocery store and in a computer shop and in a construction site? Addition is addition, it's mathematics, it works everywhere. This is why this also works everywhere, but you have to carefully tune it, and it's very difficult to optimize. Optimization is slow. So into the system, you can put anything you want in this domain or in other domains. You can put any DNA elements, EQTL, you can put expressions, methylations, copy number variations, you can put treatments, adverse effects, anything you want into the system. But each time you pose the system, it corresponds to a different computational intractable continuous optimization problem for which you have to propose objective function, optimization solver, prove convergence, correctness, and optimization is so. And this is why you need to utilize high-performance computing. I will not talk about other methodologies, but you can, if you want, this is the first paper in which we utilize this methodology back in 2013. This was when Marinka Žitnik was visiting my lab at the time at Imperial College London. She came as a PhD student from Slovenia, and after finishing her PhD, she first went to Stanford, and now she's a professor at Harvard. So this is the first time we utilized matrix factorization, and in particular matrix 3 factorization, to rewire disease ontology. because disease ontology is 100 years old, based on the symptoms and most affected organs. Disease ontology is basically how diseases relate. And, you know, if you have diabetes, for instance, are you more prone to certain types of cardiovascular disease, or does the diabetes protect you from another disease, etc.? This is what disease ontology is. But as I said, this is 100 years old. 100 years ago, we did not have molecular data, and the question is how, how would it look now in the light of new modern data? And this is what we did back then, 8 years ago now. And this is the first demonstration that this methodology works in this domain, the matrix factorization work. So you can read that as the first paper in this area. Then also we utilized it to rewire gene ontology. This is in yeast. The previous one was human. This is in yeast. Again, similar thing. And we also did a multiple network alignment. I forget in which year, 2016. So as you've seen, even aligning two networks, pairwise network alignment is very difficult, but then aligning multiple networks is also difficult, and also how to integrate both the omic data and also the wiring patterns, the topological data from the network. Again, we use matrix factorization for that. So a very versatile methodology. Now let me conclude. I hope I've demonstrated how complex and heterogeneous these biomedical data are. They are getting larger every day, of increased heterogeneity, highly dimensional, growing complexity. They're noisy, they're dynamic, they're different time and space scales. We've seen that Nature and World Economic Forum, they've identified these problems as key to resolve in the next decade. And I hope I've demonstrated that each type of these data that we have is of limited but complementary information, and this is why we need to jointly organize them and mine them within the same framework. And we have grants to do that. To holistically mine all of these data, we've seen some paradigm shifts. We need conceptual, so meaning biological paradigm shifts, and methodological as well. The conceptual one, I believe, is happening now. People are not relying on analyzing only sequence nowadays. There is an understanding that we need to analyze all types of data, and there is a realization of the new bottom-up data-driven concepts and biology we uncovered that it's governed by yet undiscovered principles of life, and that may point to ways to rethink biology and approaches to medicine. One prototype we've seen in the ISAB, but we're not the only ones seeking for this. We've seen a beautiful paper, I thought, from Santa Fe, Statistical Physics, in midsummer this year, called The Multiple Paths to Multiple Life, where they propose this concept that at the bottom you have the biological materials, the space of material possibilities. You have inorganic and organic chemistry there, principles of kinematic self-assembly, biophysical law, cell theory, molecular dynamics, protein folding, cell sorting dynamics, etc. This is all one layer. Then you have constraints on them, the architecture, the dimensionality, topology, conservation laws, and the design principles. And on the top of that, you have optimization principles. You have measures of information, free energy, algorithmic complexity. This is exactly what I talked about. And geometry, this is another thing I talked about. And basically, they came up with new biological theories in this particular paper, extending the definition, the current definition of life. Very, very interesting. I think we live in a very interesting time in the history of science. Now, to make these paradigm shifts, of course, you need methodological improvements in mathematical formalisms that will capture all of this, the scale organization, the dynamics, the stochasticity of the data. And even when those formalism exists, such as when we saw hypergraphs and spatial complexes, and there are no algorithms to compute and extract information from these models. Once you model the data, how do you compute on it? So we made some algorithms. This was just basically illustrations of these hypergraphs or simplets, etc. There are many issues to resolve, computational issues. Let's not go into that. But very importantly, we need to educate young people to these so-called embedded data scientists, because computational issues will stay with us. They're arising from mathematically provable computational intractability of these problems due to large sizes, heterogeneity, etc. And this is why the only way is to be problem specific, make these heuristic approximate methods, solve them using high performance computing. But for that, you need these Renaissance scientists that speak all of these languages. And to that end, we have published a book 2 years ago by Cambridge University Press. My lab wrote 5 out of the 13 chapters of it, and this is for graduate students, master's, PhD students in all of these disciplines: biology, mathematics, medicine, computer science, optimization, etc. Hopefully to contribute to these efforts, and this is why I love being part of this training network. I think this is exactly what we should be doing. We need to not forget that we are guided by the needs of biomedical collaborators and the industry, but we cannot become slaves to them, just servicing them, because often they don't understand what they need in terms of methodology, and this is why but in close collaboration, of course. And as you have seen, Europe really wants now in the New Horizon Europe framework things strictly out of academic settings to commercialize, to productize, and we are trying that for our ICER. Let's see how successful we are. This is a completely different world, but quite challenging. And then in the end, I would like to thank for funding all sorts of organizations that supported my work throughout the years. The work that I presented, also learned societies that recognized it. And the most important thing is to thank my group members, without whom none of this would have been possible, past members of the group, and also my colleagues and collaborators. This is when before COVID, the group, also the last retreat part of the group before COVID. It was literally 1 week before the lockdown, and this is now. And thank you very much. And I took this.
All right, thanks so much, Nataša, for this very wide view of all the things that you've touched upon. Very nice, complementary to what we've heard before in the meeting. I want to open it.
Yeah, I'm not sure what to do now in terms of, should I stop sharing or have I stopped?
It's good. Yeah, you could also stop sharing. No, don't, because people
okay.
might have questions about specific parts of the talk. Yeah. So do we have online questions or questions in the room? There's a first one from Karyophilis.
Yeah.
Okay.
Hello, my name is Karabers Karyophilis, ESR 14. Thanks for the presentation. It was really nice and interesting. I wanted to ask you something about the drugs that you mentioned, that you can use the models to predict genes interactions, protein interactions. And, I was interested to learn if you can use this model in order to propose some dosage adjustments, or to suggest for a patient, for a cancer patient, to use an alternative drug, for example. Do you have these potentials?
Yeah, we published on that. This PSB paper, Pacific Symposium on Computing paper that I referenced in 2016, is exactly what it does. For cancer patients, for serous ovarian cancer patients, we took like 343, I believe, from TCGA at the time, did all of this data fusion analysis and proposed drugs to repurpose for them. Yes, absolutely, yes.
Okay, and another question. How you deal with to fill in the gaps between the scientist and the physician? For example, you have this model, so you can propose something. Is it familiar, the clinician or the physician, in order to guide him, to for their final treatment, let's say. I mean, do you think that this is going to work for the better communication between a scientist and a physician?
Yeah, hopefully eventually we will get there. I mean, right now it is very difficult. I mean, as you're aware, the system is overburdened by this COVID situation, right? I don't know what to tell you about that in the sense, I mean, there are physicians who understand, of course, these methodologies. Many of them don't, let's be frank. But there are those who understand. Now, honestly, this is a very complex sociological and political issue. I think the fastest way, maybe, to, if you want to bring something to the patient, is to go through a startup. Make a startup, have that startup be bought by a large pharma, and then large pharma has all of these channels that are needed. But to go through institutes and research hospitals and even large pharma, just to work for large pharma, this is all too slow, very slow. This is going to take years. So, and honestly, I don't know. I don't know how to resolve this. I think there are, I mean, there's all of this stuff about the data privacy, security, about safety, about, right? Gender dimension, and many things. They're just kind of trying to orchestrate it all. And I think we're at the beginning. Honestly, the fastest way is this that I told you, startup, do a startup.
Yeah.
Because then it's not up to you, because then you're plugging into this system that knows how to do it.
Okay, thank you.
Other questions?
Hi, I'm Marina, and I have a question about data integration. So you say that the best option is to holistically integrate all available data sets. But talking about, for example, different geo-enrichment tools, they are not fully complementary and sometimes contradictory, like in a very minor detail. So I would like to ask you, what is your vision on this data sources ranking and filtering in terms to filter out false positives or some kind of other weird findings? So how do you prioritize or filter the input databases you use?
Yeah, thanks for that question. Yeah, data quality is an issue. Many people work on that. The main thing, and I mentioned that, but probably very briefly, was full of information. You need to make sure your tools are robust, your algorithms are robust. You need to test your algorithms in the presence of this noise. This is much more important than filtering your data. Nevertheless, we do filter our data. For instance, if we want high confidence protein interactions, we make sure that they're validated by multiple labs or maybe by multiple kinds of experiments, etc. We can do that too. So basically, to come up with some sort of a consensus or an intersection of what different sources tell you, then you reduce the size of your data. I'm not a fan of this. I think it's much better to utilize all the data, but that your tools are robust.
Yeah.
So make your own tools. Make sure that they're robust.
Yeah, but it also leads us to the question how to prioritize these tools, like how to find the best database to follow this particular source. Like, have you deal with that challenge already, or is it just like no priority?
You cannot tell people how database is bad, and other people you're going to make enemies. Of course, I will never do this. But some usual suspects, right? Go to where the most data is. Go to the largest ones. Go, read some recent good quality papers by good quality labs from good quality journals and work on the data they use. If you don't know, look, maybe you want to spend your life. They're different people. Me, this doesn't excite me. Filtering of the data, ranking of the databases of people. We all know about measuring, things, how much air you breathe out of your lungs. I don't care, right? If you want to spend your life on that, go. This does not interest me. Algorithms interest me. So no matter what data you give me, I want to be sure that the algorithm will, even in the presence of this noise, give a good result.
Yeah, okay. Thank you. Thanks. This is a question.
Anything else?
Hello. Thanks for the nice presentation. I'm Behnam, ESR 8. Well, my question mainly regards the graphlet part. The graphlets seems interesting to me because they remind me of wavelet transform in signal processing, but there we have a specific actually for the choice of the graphlets. Is there any specific reason why we used these structures, or as in wavelet transform we've got the orthogonality constraint that we want our atoms be orthogonal to each other?
Very, very nice questions. I really like your question. I'm sorry I wasn't more clear on this. We take all graphlets, okay? All of them with 3 nodes, 4 nodes, 5 nodes, 6 nodes. After a certain point, you don't need to go because you work in a small world. It's all small worlds. So even with 4 node graphlets, you reach everywhere. So basically, you reduce the quality of your results if you go beyond certain size of the graph. Once you have, let's say, 4 node graphlets, you don't have very many of them with 2, 3, and 4 nodes, right? All together. And then orbits, you have a little bit more, right? But the point is you take them all. So the thing is, for instance, I don't know, the network motifs, they only wanted the statistically overrepresented ones. No, we don't know what statistical overrepresentation means. As you saw, for instance, differential expression, it only tells you parts. Even the low occurrence ones have a meaning. So this is in that regard. Now orthogonality, very, very important. So basically we want independence. Now in terms of graphlets or topology, we don't have that, right? We don't have that notion, as you correctly pointed out. Is probably a way to design it. But in our most recent work that uses graphlets, but it's different, absolutely. We care about orthogonality, but I'm not going to talk about that more. I mean, we have done it. It's just that my students are currently writing a paper on it, so I wouldn't want to talk about that. Once they have write a paper, we, but excellent. You're thinking along very good lines.
Thank you. Thank you so much.
We have other questions in the chat here. Thank you so much for the amazing talk, for instance, from Farzaneh Firuzbakht. She asks, can your graph-based drug repurposing method predict new patients or new drugs?
From the particular framework for repurposing, perhaps not, but you can extend the framework, right? So predict new patients. I mean, what that means, you would have to put in people, right? So for instance, not only cancer patients, but also diabetes, cardiovascular, whatever, right? Then of course, if you put them in, you will be making predictions. Now new drugs, now this is interesting. Now this is very interesting. So you're asking whether this methodology could produce new drugs. The answer is yes. Again, work in progress that I chose not to speak about because, I mean,
Okay.
Yeah, for obvious reasons. But yes.
Right. Another question from Paddy, something, open parentheses. While integrating contextual omics data with interacting networks, we usually use interaction networks which are non-contextual. In other words, the interaction networks are derived from heterogeneous experiments, different cell lines, different contexts, etc., etc. How do we reconcile this non-ideality?
Yeah, absolutely. Yeah. Good point. So the protein interaction, this is about protein interaction networks. they're generic. They are for all conditions, everything that has ever been seen. So this is why this thing is large, right? And this is, this kind of protein interacts with this kind of protein under some conditions. So they are generic. So this is only describing what is possible, not what is actually happening at that particular time. Now, there are standard ways people have been doing this for a decade, how to reconcile that. For instance, expression data, the overlay expression. If it's expressed, then you take the sub part of this network with that. Or if it's in this cancer, if it's in this tissue, or this condition. So, and this is how we have also done it in this iCell paper that I told you about. So there are other ways how you could do it, of course, but again, gets into perhaps too much detail. Sometimes, we don't see the forest from the trees, and we are, it's also good to keep a perspective and not to dive in. So this is usually how we do it. Yeah. And everybody else does the same way. I mean, something else becomes more available. I mean, there's now more data, there's now newer data, there's now this single cell data, there's all sorts of stuff that you can, then if you have those data, then you're fine. But yeah, in this particular case, overlay something else that you're sure is happening.
All right, maybe one last question from Professor Damiana Rosman, who is a partner in TranSYS, just like you are yourself, Nataša. She writes, very nice talks, thank you. How do you manage to communicate new disease classification findings to the rigid medical community? Many think this is a must, but changes are slow.
Yeah, I have not been very successful. I publish it and that's it. Again, this is a political and sociological issue that, okay, what do I do? So, I'm involved in certain organizations such as Academia Europaea. I'm an elected member of them, also of the Royal Serbian Society of Arts and Sciences. Then you go and lobby. I mean, in particular, you go to the EU and try to lobby. Standard ways of organizing and trying to impact. Guess what is going to be how long it's going to take. I'm not overly optimistic. Things change. But mind you, I was going to say things change slowly, but mind you, we are now living in a very rapid change. This whole situation we are in for a year and a half maybe is going to speed things up for better or for worse. I mean, in England, there is, I spent a decade in England, and they say, little change is good, but no change is better, right? So there is resistance to this. I don't know. I think as scientists, we can just say our views, but I think it is important maybe to get organized and to fight for what we believe in if we would have any time. Very, very easy.
All right. Good. I think that's it for now. We need to break. Thanks a lot again, Nataša, for your talk and the nice discussions. And to the thanks again to you. To the online audience, we'll be breaking now for an hour and back at 1:30 p.m. European Paris time, let's say, and we'll see you back then for Johan Dreo's talk. Thank you, everybody, for attending, and see you soon. Bye. Hello, everybody. Welcome back to the TranSYS Data Analysis for Personalized Medicine meeting. I don't even know who's online right now. I'm guessing it's both the next speaker and I. Thanks for coming back after lunch, or wherever is appropriate in the time zone that you're in. Our next speaker is somebody who's significant experience both in academia and in industry, and he's going to talk about certain tools and technologies that are important to make all of these, let's say, more theoretical and new concepts a reality on this, especially on the side of software. So it's my pleasure to introduce Johan Dreo. who's also a new member of our group in Pasteur here, and he will discuss interesting stuff that I forget the title of. I'm sorry, Johan, he's sitting opposite to me. I'll pass it on to him, and I hope you enjoy the talk. Thanks, Johan.
Thanks, Benno. So the talk is going to be about practical tools for ensuring reproducibility. So let me do that. Okay. So the idea of the talk is to present a list of practical tools that you can actually use and how they are improving reproducibility. So reproducibility, I guess you heard of it. It's something that's crucial for science and it's very often overlooked. And I will introduce a curated list, not all the tools that exist on the market, but the ones that I've found to be the most interesting. So why reproducibility and what does that mean really? So in the past years, there have been a lot of debates in the scientific community around epistemology about this so-called reproducibility crisis. So we should all know that the scientific method is about falsifiability, so about empirical evidences, about experiments, and about predictable outcomes of experiments, so that we can build up cumulative science and go forward on the shoulders of the people that works previously. The problem with that is that, as is shown by several publications now, but I've selected this one from Baker that has been introduced in Nature, is that in many occurrences it's difficult to reproduce an experiment. So most of the researchers that were interrogated online here have failed in an attempt to reproduce an experiment. And worse, they have failed to reproduce their own previous work. So if you're new or quite new to science, you can think that seems strange, but we have all been beaten by that. It can be very hard to reproduce even your own previous work. So reproducibility is a term. So there have been some try to design some terminology around that. So I've selected this very simple one. We think about repeatability when the same team is doing the same experiment again and expect the same outputs. Reproducibility, on the contrary, is when a different team is doing the same experiment. And of course, as I said before, a different team can very well be yourself, but the years after. The next step is replicability, when it's a different team that's doing a different experiment, but trying to answer the same question. Okay. And there is some idea that I like that go even further than that, but I have no time to explain that in details right now in this presentation. It's called generalizability, and I've put the reference in there for you people that would be interested in that. So to improve reproducibility, there's a lot of action that we can take. The first set of actions are what I call here cultural actions. So one that are not really tied up to technical action that you would take yourself. So of course there's stuff like publishing negative result. If you fail to reproduce a study that would be interesting for the others, do not oversell positive results. fight bibliometry, do better statistics, of course, and although refrain from hypothesizing after the results. And as I'm going to present in this presentation again and again, advertise testing. Okay, so I'm not going to discuss those cultural aspects. I'm going to try to convince you that there is tools that can help you. So what I call here technical actions. It comes in a nutshell to it falls down to a very simple idea, which is you should automate everything. Because automation, it's a good way to ensure that you have a non-ambiguous description of what you're doing and that others would be able to read and execute. So it comes in 3 sections. First is it's doing backups, what I call here keep track of artifacts being source code, data, or whatever. So for that, we have a couple of tools. We have version control. We have the electronic equivalent of laboratory notebooks. And we have archive tools. We have some tools that help to do good engineering. So I will discuss that. You should do modular code. You should do test. You should automate whatever you can. And that would help your future self if you do that in a reproducible way, meaning if you have a description that's easy to read and execute. So we are going to see containers, pipelines, design of experiments, and publishing. So let's go into the details now, so the tools themselves. So the assumption I made while presenting this is that you know how to read and search documentation. I'm not going to explain everything about each tools. It's rather an enumeration of the tools, and most probably you would have to go and follow the links that I give to understand more precisely what it's all about. But in 1 hour, I don't have time to do more than that. And the idea is to solve this difficulty, which is very often we don't know that a tool exists. We don't know what to search for. It's difficult to find, and sometimes we don't even know that we can do that. So this presentation is here to solve that. And whenever I had several options, I chose the one that involves Python because it seems to be a language that everybody likes and most of people know. So this presentation is this curated list of tools. It somehow insists on testing, as you will see. And I nevertheless gave more alternatives or more information in the See Also section that you would see display, but that I will probably not comment very much. But the presentation would be available after, and you can download it and you can then follow the links more thoroughly. So the first set of tools are tools that implement version control. So here there's a tool that dominates all the other, which is called Git. And Git, as a version control system, is able to keep track of any kind of change that you would make to your code. So the idea is a little bit like making incremental backups on top of each other's. So that you would have the ability to, for instance, go back in time. You would say, I've made that change, but I'm not okay with it now. I would go on the previous state and then do something else. So this is really useful. That helps debugging, for instance. That helps working with others on the same code base because there is this ability to do what we call merging. That is, take source codes from many other people and automatically merge them in the same code base. And the key word here is being automated. It do a lot of stuff for you. So instead of copy pasting, you would just say, please get this code. Git also have this interesting property that it works offline, and it's very well set up to work offline and still do backups online, as we're going to see. So as for reproducibility, the basic idea is that all the source code is versioned, even the test that you're going to write, and you're going to write test. So every source file, meaning not the generated file and not the data. So everything that is considered as a source code. And in the vocabulary of version control, you have a state of your code, you're doing a modification, then you commit it, meaning you register it in the repository, in this history. And then you merge these commits into some version. So I pointed here, there's a lot of tutorials, of course. I point here, there's some tools that are useful. So the first one is a short tutorial that propose what we call a workflow, which is very common with Git, which explain you what you should think when you're coding and committing your modification to your source code. GitK is the classical graphical interface toward Git. If you like to use the terminal, and most people seems to prefer graphical interface, but usually when they try the terminal, they find it more easy, surprisingly, but that's kind of true. They find it easier to use Git. And this tool, Liquid Prompt, is a way to show the state of your Git repository right within the terminal where you would do commits and do Git commands. Okay, now that you have a Git repository with your source code versioned, and one thing that you can easily do is to do online backups of it. And there are even commands in Git to do that, which are push and pull. So push is to push the backup online, and pull is to get the backup on your computer. So the website I propose here, it's GitLab, and it comes on top of allowing for Git backup. It comes with issue trackers. So if someone, for instance, see a failure in your test, then they can tell you in that's basically a to-do list, and they can tell you, okay, I have found this bug. What there is also this kind of website that they allow you to merge some code that will be proposed by some other people. So what we would have done before is that the people would have sent you an email with all the code saying I fixed the bug, but you don't know where exactly, you have to copy paste and so on. With this kind of service, it's very easy. You just have to push a button and you would take the code that the other proposed. They also provide basic public page hosting, which is nice if you want to show documentation or some explanation, and it's right there where you are working, so it's handy. The alternative to GitLab, which is very well known also, is GitHub. I've heard of it. For those of you who are already comfortable with that kind of website, I really advise to have a look at what we call now DevOps, which is a fancy name for everything that goes around automating, building, packaging, and testing of the software. And GitLab proposes a lot of features around that. Okay, so now you have your source code somewhere. You have something that starts working. One thing that's interesting, it's the equivalent of laboratory notebooks for those that knows a little bit of wet labs. And the idea is that you have a page, a web page actually, which is a sequence of paragraph of text and executable codes that produce figures, for instance. So it's a very good way to keep track of your ongoing pipeline or experiment or what you have tried, actually. It's kind of easy to share with others. That's a single archive that you can pass over. And it's very good for showing tutorials or demo of your software. It can also, to implement demo, what you would do is, for instance, you would show the test that you have implemented. And your notebook would use the software as a backend. So you can put that online, and there is an online demo, and there is a hub that serves notebook on which you can publish already done notebooks. Okay, so I said in your notebook you should use test to implement a demonstration, but what is test really? So I found out that it's kind of obvious for most computer scientists or engineers, at least. It's now common practices to do a lot of tests, and I found out that for scientists it's less common, but that's really important. So there's a few golden rules for software engineering. The first one is that you should code modular. So you should not put a single page of thousands of lines of codes doing something. Every time that you can, you should put a fragment of code in a function. And at the end, the set of function, we call that a module, as in Python, for instance. And functions are kind of the atomic objects of programming. And the idea is that you're going to use that, this fact that you have some atomic chunk of your code, you are going to test those chunks, so those functions. And to do that, the first line of defense are asserts. So asserts, it's a function that comes with virtually all the languages that would make the program crash if the predicate that you pass to it is failing. So we're going to see some example of that. You should put asserts right at the beginning of functions, which we call asserting preconditions. You should have a look at the arguments that you take to this in this function and check that they have the good properties, the properties that you think they should have. Even if when you're coding it makes sense, it's obvious to you, but believe me, in 6 months it's not going to be obvious to you. So it's very good practice to double check that. And in the same way, once you've computed something with your function, you're going to return it. Before returning it, you also assert that this data structure that you're returning has the good properties. And we call that asserting post conditions. Basically, and that's the second rule, you may put asserts everywhere in the code. That's good practice. Once you have a function with asserts in it, and you can do unit test. Unit testing in the engineering vocabulary means that you have this function and you're going to pass it simple data on which you want to ensure that it passes, it computes something that's correct, or it fails. For instance, if I give an array of size 0, most probably your function expects something in the array. So if I pass something that has no size, of course it should fail. So those 3 or 4 first line of defense up until unit test, you should really do that. That's mandatory. You cannot avoid that. The thing that's optional for most scientific study in bioinformatics is maybe integration tests, which is what you would expect from real data. Okay, so I would really advise you to do it. But if you don't, I'm not going to fire you either. Whether I'm going to for if you don't do unit test. And the third rule is that once you finish to code something that seems to work, you've added a feature to your software, to your pipeline, a step to your pipeline, you should implement tests and run them. And every time you do a feature, you should run all the tests, because it's very probable that you've touched an area of the code somewhere within the dependencies that maybe changed something, and that changed some assumption that you would have had on other portion of the code or another step of the pipeline. So every time you complete a feature, you run every test that you have done. So it looks like that in a code. You have this function, so that's Python. You have a function, it takes an argument, which is expected to be a list, so it has a len, and you would check, for instance, that the len is between 0 and 100, whatever. You would check that every number in it is non-null, for instance, that kind of stuff. You can also do asserts on intermediate variables. So here I copy from one array to the other, I expect them to have the same len. And you assert on the result. So that's precondition, postcondition, asserting. The basic unit test is in an, so maybe separated executable. You would pass some argument to the function for which you know already the results. So here it's computing the sum of the list, of course, so you know that with this argument you're going to have this output. So that's the very basic unit test. And you can also test, at least in many languages, at least here it's very simple implementation in Python. You can test also that test fails, actually, like that. So here we are catching the assertion error, which is raised up by this assert function. And if it failed, then we are happy. And we assert it again, so as that if there was no assertion raised when calling f, then we raised in the test. So it's interesting to note that, okay, you would say all those conditional branching, they're going to slow down my code. And that's true. If you do a lot of asserts, the code is slowing down a lot. But the nice property of asserts is that in almost all the languages I can think of, you can declutch them. You can say, for instance, to Python, if you call Python with this -o option, instead of just calling Python right away, it would remove all the assert code. And it would remove all the tests that are below if debug, this global variable here. And that's going to execute a lot faster, of course. So you have what we call the debug mode when you're coding and you want to do the asserts. And there is the release mode where you want to execute with maximum speed. And you can do both with a single code base, which is the point. So in a nutshell, the test helps avoiding bugs because you would have an error if you would have one, of finding bugs, of fixing bugs, because if it fails, if an assert fails, it fails right there where you need it. It helps doing that on the long term, because the test that you wrote at the beginning of your application are still going to be valid after. And if you build up your tool on top of each step, then the low-level steps are going to be tested even after. And despite what you could think, it actually helps you coding faster, because debugging something that is not modular and not tested, it's really a nightmare, and it can take a lot of time. Whether if you did that, you're going to know exactly when and how things are going hairy. So there's also a nice feature, especially for pipelines, is that you can, using tests, double check the intermediate data. If your pipeline is a sequence of steps like that, at each step, you would have wrote the test, okay? So those tests, they produce something that you can look at to double check that, for instance, with new data, all the intermediate data that you produce, they looks good. So in R, there is this function that's called stop if not, and there is many packages that are assert in their name that you can use to do that. There are also basic unit test modules. So in Python, there is this unit test module. And if you want even more, there are what we call frameworks that automates a lot of stuff and helps you describing precisely what kind of test you do. So in my opinion, you should at least use if you do Python, you should at least use the Python unit test. Okay, and about this DevOps thing that I mentioned earlier with GitLab, what we call continuous integration, it's really the fact that on GitLab, you can tell GitLab that on every commit, for instance, every time you push something on GitLab, it would run the tests for you. And after that, if there is some test that's failing, it would warn someone, so the author of the commit, most probably. It would send an email saying, okay, you've pushed something on your software, but it's failing the test. So that's very handy. Okay. So now we have a code base that works, that is tested, that is backed up online, and we would want the other to use it, right? But it's not that easy to build a software, for instance. So there's an affliction in computer science, which we call usually the not built here problem, which is, okay, it's obvious for the author how to build his software, but it's not obvious for everyone, right? So we need something to ease that. There's also another plague, which is the dependencies. Most probably all of you have faced that already. It's when you want to use a software that you did not make, for instance, you have a module that you need. It has been made by someone else, but the version which works with your code, it's a precise one. And itself, this dependency depends on other dependencies with very precise version, most probably, and so on and so forth. And how do you ensure that you get the good version with the good dependencies? And that's, if you do that by hand, it really becomes a nightmare very rapidly. There's also the problem of backward incompatibility, which is you've made a software that works with some dependency, but then for some reason the author decided to change the API, and when you update the dependency, nothing's work anymore. So how do we ensure that we can give to users a simple way to get the very same environment with which you've developed your software? So to do that, we have several options that we call virtualization. And they came historically with first virtual machine, and then I'm going to present you the other options. So virtual machine, they try to replace the computer, the hardware, with software. So basically you have some kind of emulator of the processor, the disk, and so on. So basically it allows you to run any operating system on any other operating system, which would run the virtualization, the virtual machine. So that's a bit slower, of course. And one of the main drawbacks is that it takes a lot of space, because you would have to install, to do a virtual machine, it's an installation of the whole operating system. And usually it's several gigabytes, right? So you would need to do the installation, run it, then do some more installation on it, and then start developing. So the tools that I propose here it's VirtualBox, and maybe you've heard also of the commercial concurrent, which is VMware. So this problem of having the very same environment can also be solved instead of giving you a whole computer, which is what the virtual machine would do. You can be very precise on the versions of the dependency that you want to have. And you want to ensure that you can install several specific versions on the same computer. So that's exactly the problem that solves the package manager and the virtual environments. So the package manager are here to allow you to install very simply a software and its dependencies, and possibly with a very specific version. And the virtual environments, they give you the possibility to install many versions of the same software on your machine. And you would decide at runtime which one that you run. Right. So this tool, Conda, is very well known, I guess, in bioinformatics now. And historically, it was made for Python, but now it works, or it's supposed to work for many languages, and also for R, for instance. So with Conda, you just have to distribute the configuration of your environment. So the list of software that you need, the version of the software that you need, and that's all. You don't need this complete machine to do that. So there is a repository that's the version that's specific to bioinformatics, which is called BioConda, and there is MiniConda, which is a very simple to install version of Conda. So one of the problems that we faced with Conda is that, yeah, you have the configuration for your environment, but it doesn't really work easily for any kind of operating system. Very often it's easy if you are on the same operating system, but if you change it because the package manager, they would not have version for all the OS, for instance, it can be a little bit difficult. So there's no some kind of compromise between virtual machine and package managers that became more and more important in the industry, and they became standard actually in the web industry, for instance, and they are called containers. So containers, it's virtualization at the operating system level. So you do not abstract the machine, you abstract the operating system. So that makes up for lighter files once they are installed. You don't have to bring up the whole operating system, you just have to bring up the layer on top of it. But still, you can run any software on any operating system, on any host. So it's a little bit faster than virtual machine. It takes a lot less space, and it's a little bit more stable across operating system than virtual environments. So what you actually do is that you describe, you implement a script, which is a list of the software that you need to install to run your own software. And then automatically these tools, they would start from a fresh operating system, for instance, and they would install something on top of it, the dependency that you indicate. And then you can give this container, this built container, to people that would need to run your software, and it's very easy to do so. So of course you can also run the test that you've made within these containers, and that's actually a good idea because it gives you a good feedback on what a new user would face while trying to build or to install your software on a fresh machine. Because very often when you're a developer, you have installed a lot of tools, you don't recall exactly all the dependencies on all the installation step that you've taken already. But if you try that in a container, you start from a fresh install. It's very easy. And then you can very easily list and test which version works, which version don't work, what are the dependencies and so on. So for this tool, I advise to use Singularity. And you may have heard of the historical version, which is called Docker. But I prefer usually we prefer Singularity because it's able to run easily on a cluster. And in bioinformatics, that's a very interesting feature, right? So now you have a container with your software. It can run easily on the cluster without bothering the sysadmin with installing dependencies. You have installed them in the container already. What would be nice would be that this pipeline that you're making, it would be easily described in a non-ambiguous way in a script. And it would be nice if this script can do the parallelization and the job submission on the cluster by itself, right? So if you've never used a pipeline, or sometimes it's called a workflow manager, you surely have some script somewhere that calls every step of your pipeline. And most probably, you may have used some parallelization somewhere for single machines, but that's most probably the most you've done. If you describe this script with a pipeline management system, it would do that automatically for you. So what you actually write, it's each steps, the script that you need to call, and what inputs this steps is taking and what outputs this step is giving. And you just describe each steps like that. So that's a way to describe a graph, right? A direct acyclic graph that the pipeline management is able to interpret so as to detect which step can be parallelized, for instance. And you just describe these steps, and then you tell the pipeline manager to run, and it will do the job submission on the cluster for you. On as many cores or as many slots, for instance, you would need, or you would tell it. So this pipeline management, so here I propose to use SnakeMage, which allows for scripting in Python. They work very well with Conda and Singularity. So you can just tell in the pipeline script, you can say, okay, I want to install something, just grab this container here, or just build up a container by installing this version of the software and so on. So there's another one that's quite well known in bioinformatics, which is called Nextflow, and you can try it out. So once you've done that, almost everything that you've prepared, if you're doing a pipeline, is automated, right? And you can give the pipeline definition to someone else. Is going to be able to run it. So that's a very good, non-ambiguous, rational way of describing what you're doing. Instead of, for instance, if you would have done that by hand, you would have write a README and you would have forgot which version, which step comes after which other, and so on. So it's really useful for reproducibility to be able to describe that in a way that it can be run. Okay, so now you have a pipeline, and the next, I would say, natural step, but that's not a step that's very well known, I would say, now in bioinformatics. So I guess that's the next one that's going to be fancy. It's managing the design of experiments. So most probably all the steps of your pipeline, they are involving some algorithms, or some function which takes some parameters which are in a way a configuration of your pipeline. And one of the questions in experimental science is, how are those parameters affecting the outcomes? So for instance, the efficiency of my pipeline. And to do that, we can automate here and though the design of experiments so as to check that. So for instance, the very basic way is to enumerate all the parameter space to understand, for instance, what is the efficiency of your pipeline regarding those parameters. So that's, for that you need to use sampling method. Another way that we may use that is called calibration, which is, for instance, you have steps that try to simulate reality somehow, and you want to optimize the parameters to come as close as possible to reality, to measures. And that's calibration. You can also do sensitivity analysis, and if you're using this tool, OpenMole, you can also do job submission in the cluster. So here, very often in design of experiment, of course, if you're doing sampling, you're trying your pipeline, for instance, with many different configurations. Of course, that's embarrassingly parallel, right? So it's very handy to have an automated job submission. You just describe which parameters you want to enumerate or to try or to optimize, and it would run on your cluster without you having to bother very much with it. So the automation of design of experiment is very well known in engineering, and there's a lot of commercial software already, very much often in mechanical engineering, though not very much in bioinformatics. Okay, so with that, you've almost automated everything that you can, and that's very nice because you have a reproducible experiment. And now I'm going to talk about what you can attach to that, what metadata you can bring, and all the way you can do that more properly, more cleanly. One small tool that's handy, it's called Cookie Cutter. And Cookie Cutter is a way to prepare project templates for you. So if you want to do a snake make a pipeline, for instance, you can use Cookie Cutter, as you see here. You can use it to dump a directory hierarchy of files and so on, of all the stuff that you need to program to do a snake make workflow, for instance. So it's very handy to follow best practices, because very often you don't remember from the top of your head what configuration file you need, where you should put it, and how that would be useful to do it the more easily. And it also avoids you forgetting important features. So there's a lot of templates that are readily available from Cookie Cutter, and you can just search on GitHub for them. And once you've done that, you can just call Cookie Cutter. This is to say that that's on GitHub, that's this user, and that's this workflow. And it will automatically create the directories and file for you. Okay. So once you have something that works, one interesting thing that you may need or you may want to publish it. But code it's covered by a slightly different legal system than writings, for instance. So the writings and diagrams, the author's right, they belongs to you. But the main difference is that the source code, the rights, they do belong to your employers. By default, you give the rights when you sign a working contract. And so it has to be a decision to publish source code of your software, a decision that you should take in accordance with your employer. And of course, it should be easy to publish source code, but sometimes you're not allowed because you would have information disclosure authorization to get first. When that's done, to publish a source code, you can just put it on the internet, and then nobody would have the right to use it. But most probably you want to ensure reproducibility by explicitly allowing users to actually run your code and do something with that. For instance, reproduce the experiment themselves. To do that, you have to grant them some rights. The first one would be the right to distribute your work, of course. So take the source code, give that to a PhD student for them to use, to try to reproduce. Most probably, they would need to do modification to fix bugs, to change some parameters. Most probably, they would want themselves to distribute the work, so you need to grant them sub-licensing work of derived work of the modification they've made. And most tied up to software, you may need to give them the right to link, so to use your work as a library. That's the meaning of linking in the sense of software engineering. And also, you would need to give them patent grants if your code involves patents. Most probably, they would need to have the right to this patent so as to actually run it or use it in their own software. So these kind of freedoms are granted by free software licenses, and they guarantee by construction that you would be able to use, to study, to redistribute, and to improve the work. And these freedoms are necessary for reproducibility. And they are even more necessary for replicability, you know, this level on top after for other teams and other experiments. So the right to distribute the work, it's essential. We cannot ensure reproducibility without it. Modification and sub-licensing, they are necessary to do reproducibility. Linking and patent grant, it's important, but in some cases you can avoid it. And the trademark grant, it's optional. So there's a lot of free software licenses. I selected 3 of them for you by the level of permission that they allow to the user. So the most constrained one, so the one that enforced those liberties. The first one, I've selected the Affero GNU public license. The second one is for libraries. So I should say that the first one, the GPL, there are copyleft license, which enforce people that use this code to release their code if they distribute their software, to release their code as GPL covered code also. So the proponent of this license would call that copyleft, and the opposant would call that contamination. The lesser GPL is here to allow the GPL to cover the library, the code of a library, but not the code of a software that would use this library. And the Apache license, they are even permissive. They allow you, for instance, to get the code and basically do whatever you want, just you would have to allow the other to use the code. But they do not enforce the code that would, the license that you would need to use for your own code. So that's for source code. For any other generic work, the simplest license are the Creative Commons one. And that can cover, and you've surely already seen those licenses in publications, for instance, or diagrams or pictures. And they are basically made for everything else. If you don't know which one to choose, there is this chooser on the Creative Commons website that helps you. You just say, I want to do this and that, and it would tells you what license you need to have. And of course, for the source code, you absolutely need to check with your employer if you have the rights to publish open source code. Okay, once you're ready with that, you most probably all of you know preprint repositories. And reproducibility needs that because we need to have access and easy access to existing publication. So reproducibility needs open access, and preprint repository are a good way to have that. Okay, so now we have published source code, like on GitLab. We have published the publication on a preprint repository. But what about the data, for instance? And what about versions? And how do I link the code and data to a publication? Well, you can do that on specialized websites like Zenodo, which allow for long-term archiving of code and data, which would be associated explicitly with a publication. So on Zenodo, you would archive the code along with the test that you've made with this code, of course. And you would link to a Git tag on GitHub or GitLab. You would say this archive corresponds to this state, this code, which is published there, for instance. You would also be able to archive data with it and give link to publication. You would say this archive corresponds to this publication. And from within the publication, you would also be able to link to the archive data, because every archive on Zenodo allows you, or is tagged with a DOI, and it's then very easy to cite, and it's a good way to have a long-term link toward this data. On top of that, Zenodo allows versions. So in your publication, you would be able to say, okay, I've used this archive version 1. On another publication, you would be able to say, I've used this archive version 2, which, for instance, fixed a bug or something like that. So there's another website which is called Software Heritage, which do quite the same for software only. Okay, so as a conclusion, just to give you a mental image of what you would do with all those tools. If you start from scratch, you would start with a small notebook, coding a simple proof of concept. Once it works, you say, okay, this part of code is a function, and you would put that in a module, and the notebook would call the module instead of just the plain code. And you got this module, and you version the notebook and the module in a local Git repository. Of course, you add asserts, now that you know that it's mandatory. And when the function works, you write the related unit test, of course. And you modularize your code like that. At the end, the notebooks eventually became a tutorial on how to use your code, because you've picked up the code, you've refactored, as we say. When a feature works, you said, okay, now is a user able to use it? You can test that, your module in a container, to check how easy it is to install. Within the container, you install the dependency with conda, so that it's easy for you to indicate which version and how it should work. And once you have that, then you have a container that you can use somewhere else, like on a cluster. So you can write a pipeline script that calls this container, and that would run the whole experiment. You try it, it works, you're happy. You can push that on GitLab for the other to fix bugs, for instance. You archive the code and data that you produce with your reasoning, your pipeline on Zenodo. You write the publication, you cite the archive, you put the publication on a preprint archive, your publication got accepted, you're famous, and you're very happy. So if I summarize in a diagram, the application management, the software part, it would look something like that. Sorry. The everyday work is done here on the Git repository, on which you code your module, which it's a set of steps for your pipeline, for instance. Those steps, you can also call them from the notebook, which is a demonstration of how to use your software. With that, you can write a pipeline script calling those steps. You can write a container definition script, which tells on which actually shows how to install your software. So you can, from the Git repository, just launching the container definition script, you can download the Git, so the source code from Git, build the application automatically in the container, install the dependencies, and then you have a container that you can just copy on the cluster, and then your pipeline script will call that. Right. So if you need so, you can publish your Git repository on GitLab, you can publish your notebook on Jupyter Hub, and you can publish your pre-built container on Docker Hub, which is very handy because the user can just download it, for instance. So what you would do there, for instance, in your pipeline script, you would say just download this container from Docker Hub at this version, and everything would be fine already. Okay, so this is a recall of the essential tools that you may need to know. And yeah, I have finished. If you want to read more about reproducible pipelines, there is a very short publication that just got out a few time ago. And do not hesitate to test your code, of course. Thank you very much.